Welcome. My name's Matt McFarland. I am the Vice President of Commercialization and Industry Relations at Nationwide Children's Hospital. I work with the Office of Technology Commercialization, and I am delighted to have you here today at our 2024 Annual Technology Showcase. For those of you who have been to this event before, welcome again. And for those of you who haven't been here before, we're excited about this event every year because it's a wonderful opportunity for us to bring together a lot of diverse stakeholders that our office and our institution work with all the time. So in the audience today, I think total today we had 170 registrants between online and in person. And we're looking to try and follow up, and some person in our office follow up and engage with you at some point during the day today. So thank you for being here and for being a part of this. Before we get running with the run of show, which is really going to be 6 different technologies that were born out of Nationwide Children's Hospital, we've tried to come up with a technology that's representative of the therapeutic space, the med device space, the digital health space, some of the different areas that we're working in. We're going to have an opportunity to work through and have you hear from some of our inventors and some of our companies that we're working with. And then we also try to, at this event every year, focus on a topic that is of interest in general to the group that we've assembled. And so we've had keynote speakers before, we've done panel discussions, and later today, I'm really excited about a panel discussion, kind of like a 5-person fireside chat we're gonna do with people associated with the Midwest Pediatric Device Consortium and pediatric device consortiums in general. Here in the U.S. So you'll learn a little bit more about that as the day goes on. But before we get rolling, I want to take just a couple of minutes to provide all of you, whether you're new to our office and you need an introduction to kind of who we are and what we do, or whether you've been working with us or familiar with us for a while and it's just been a while since you've been updated. I'm just going to go through a few quick slides to kind of give a snapshot of where we've been able to do some effective translation here at Nationwide Children's and where we think we're marching on some different fronts in the future. This first slide I have up, it's important to highlight that, our hospital system culturally stays very focused on our strategic plan. Every 5 years, we work together as a hospital system to come up with a strategic plan that can help guide all of our actions and our efforts when we execute in our jobs. And our current strategic plan is called Leading the Journey to Best Outcomes for Children Everywhere. And I know that's really broad, but the impact we have is broad, and it takes a lot of different inputs to make that happen. And for the OTC, where our input in really is summarized at the bottom of this slide under the circle that says partnerships. And really for OTC, that's the essence of what we do. Our mission statement is guiding ideas to solutions. But the way we do that, we're never going to sell a drug here at NCH or be a biotech company or sell a med device. So we have to partner. We have to partner our technologies out with organizations and for-profit entities that can do that. And we also have really tried to become more robust in taking the translational infrastructure that we've developed here at NCH and figuring out how we can partner that with technologies that aren't ours, but that could use the help getting to a place where they're impacting kids. And so, we do that through a process in our office that I'm not going to go into this or spend a lot of time on it, but for those of you familiar with tech transfer, we're a very soup to nuts operation, and that if you are an internal customer inventing something, or you're an external investment group or company or startup that we're licensing to, you're probably going to have one individual, one licensing professional here that you work with that's your main point of contact. And we staff the rest of the office in this way. We've grown a little bit in the last few years, and we're now up to a team that, depending on how many interns we have in the office that month, we hang out around 20 people. Dr. Margaret Barquette leads our licensing efforts with a team of licensing professionals that all have the technical chops to dig in on the technologies we see and the business acumen to facilitate deals. And then Susanna Woolman is our operations business manager, who works with all of the other professionals in the office who maybe aren't as deep into the technical aspects of tech transfer, but really helps support all the other functions. And I think it's important to recognize this team and all of the hard work they do day in and day out for Children's, and also putting this event together today. So if you're here with the OTC today, could you stand up and maybe we could give you a round of applause for all the work that you do for commercialization here at NCH. Thank you for your efforts. It truly is is fun and humbling to watch the efforts of this team as they kind of mature and are realized. Quickly familiarizing you with the types of innovations we see out of NCH. We see a lot of therapeutics, a lot of biologic therapeutics. Gene therapy has been a big space for us, and if you're familiar with our portfolio, you're probably aware of that. We have a robust tissue engineering operation, cell therapy in the therapeutic space, a lot of typical research clinical tools, biomarkers, and then there's this category up here we've labeled end user innovation, which eventually is probably going to have to bifurcate and get a little more detailed to it, but that's where we kind of group, digital health and med device technologies that often come from our practitioners, our clinicians, or our people that are running operations within the hospital system. This is just a list of some of the startups we've been excited to be a part of launching over the last few years. You can see some of the different areas of focus that I mentioned earlier are represented here by these startup companies. And also, we have a number of companies. This is not a comprehensive list by any means. These are more the frequent flyer companies, but a number of strategic partner companies where we've engaged in several agreements and several collaborative efforts with, and over time developed a relationship in that regard. But for us, one of the things that is kind of the moment for the office to get to feel like we're standing close to the top of Maslow's hierarchy of needs is every once in a while we're lucky enough to get something actually through a regulatory process, approved, sold, and being used in patients and impacting patient health. We have a few different areas where over the last few years we've been able to achieve that. In the therapeutic space with Zolgensma, which is now sold by Novartis. There's a gene therapy for spinal muscular atrophy. And then just this past summer, our partner Sarepta Therapeutics was able to launch the approved product and go to market with Elevidys, which is a gene therapy for Duchenne muscular dystrophy. We also have a couple examples of medical devices that have been translated and are impacting patients now. The device you see on your left is the e-suction device, now sold by Olympus, but it's a device that's really specific, believe it or not, for removing hot dogs and bratwurst from the esophagus of kids. There wasn't a good tool to do that, and we had a clinician here that recognized there was. And on the right, the Comfort Collar, which is a product that came from NCH clinicians that recognized that pressure ulcers with tracheostomy patients was still a really, really big deal, and they had some really innovative approaches to materials and setup of a collar that's now sold by a company, and the sales really have increased in the last year, being used nationally now, and having a dramatic impact on this category of preventable harm in pediatric patients. So those are all very exciting things for us. Just to quickly familiarize you with, we have a lot of different resources here at NCH we bring to bear to try and create some of the pipeline that you're going to see today and you may hear about from us. We have internal funding mechanisms like the Technology Development Fund, which is basically a pre-license accelerator fund. We deploy, between $600,000 and $700,000 a year in that regard. We work with Ohio's programmatic opportunities from the Third Frontier in the Technology Validation Startup Fund. It's a source of funding for technologies that are just about to cross over that threshold into being a startup, and this is a way to help launch them with funding from the state. And then I'm very proud to claim that our leadership had the vision and the willingness to support these efforts several years ago when we started investing in early company pre-seed, seed-focused funding through mechanisms with our partner Rev1 Ventures, who's managed these funds for us, the Life Science Fund, the Catalyst 2 Fund, and the Catalyst 3 Fund. They, in order, went $5 million fund, $15 million fund, $30 million fund, and it's important to note the Catalyst 2 and 3 Fund are also done in partnership with The Ohio State University. And so between Ohio State, us, and Rev1, we've been able to put together a pretty robust network of funding the pipeline of NCH and OSU technologies that we're excited about. I've talked about a lot of this up top, but excited to report in the last 5 years we've executed, and a few more have happened in the last few weeks, so this is actually an old number now, but more than 130 new deals. We have 21 active startup companies. I talked earlier about the approved devices, but at any given time we have close to 30 different technologies, both med device and therapeutic, that are in clinical trials with one of our licensing partners. And importantly to all of this, we've been able to start to generate a little bit of a consistent revenue cycle for our technology commercialization operation. Over the last 7 years, we've averaged around $35 million in annual revenue. That's important because it's part of this kind of virtuous cycle where if we want to continue to build research infrastructure, continue to grow, continue to invest in some of the mechanisms I described before, we have to have that. Another important note I want to highlight to you today, and it is a nice prep for the panel we're going to have a little bit later on. At NCH, we recognized a few years ago that we were not really proactively addressing, supporting, and resourcing clinicians who want to engage in the med device and the innovation space. A lot of what I was able to show you earlier today is the result of clinicians that are leaning in well above and beyond their day job and kind of self-training themselves to innovate and be design thinkers and so on and so forth. And about a year and a half ago, we were really fortunate to secure Dr. Corey Chris as one of our new faculty members here at Nationwide Children's Hospital, who's an excellent pediatric surgeon for us, but also leads an effort called the Nationwide Innovation Center, which is going to kind of fill that gap of educating the clinical side of the house, being a resource for them to innovate with, and an infrastructure that supports their thinking through problems they'd like to solve, and we think is going to really create a nice virtuous cycle with that demographic as far as growing our med device pipeline. And then what we'll talk about a little bit more later today is not an NCH initiative at all. We're part of it, and we're honored to be a part of it, but here within the state of Ohio, we were fortunate over the last year to secure federal funding from the FDA for a pediatric device consortium. Ours is called the Midwest Pediatric Device Consortium. And so it's a collaboration between a lot of different entities in Ohio. The academic collaborators on it are OSU, NCH, Cleveland Clinic, and Cincinnati Children's. And so excited to talk to you a little bit about that later today. And finally, before I get off stage and get the party rolling, I would be remiss if I didn't point out, and kind of bringing this full circle to the first slide, we really do believe partnerships and collaboration is key to success of any form on this front, and we really value our partners. Each of the, and this isn't a comprehensive list either. There's a lot of groups we're starting to partner with more, we're learning to partner with more, but this is a small inventory of some of the groups that have really been a robust partner for us and that we communicate with regularly, and we really appreciate the support and some of the shared programs we're able to participate in with them. So with that, we're going to get things started. Of the 6 presentations today, 5 will be live, 1 is recorded, but I promise you it's recorded because the company found out they had an opportunity to go be the keynote at a national NICU innovation conference, and that's a really huge opportunity for the company. We would not want them to miss that or not be a part of that. So we also didn't want them to miss sharing with you today. So they've recorded their message, and to introduce them, we have Andrew Corus, one of our senior licensing associates. Thank you for being here.
Thanks, everybody. My name is Andrew Corus. I'm a senior licensing associate here in our Office of Technology Commercialization. And it's my pleasure to introduce our first presenter. Dean Cook is the founder of Smalltalk, a company that came out of NCH licensing a medical device for the use of speech-language acquisition in premature infants. After licensing it, they've spread out into secondary language acquisition as well. And as Matt mentioned, they're not here today to give their remarks personally, but they were invited to be the keynote speaker at the Gravens Conference in Florida, so we appreciate that they were able to do this and that at the same time. Afterwards, I will be available for questions and answers as well.
Hi, I'm Dean Cook, co-founder and CEO of Smalltalk, where we are focused on brain development through early language exposure. My co-founder is Natalie Maitre. She is a neuroscientist and a neonatologist that I met here at Nationwide Children's Hospital. Now, we share an interest in prematurity. My background, as you'll learn, is predicting and preventing preterm birth. Natalie's work as a neonatologist is in improving outcomes in premature infants. What you'll see is that we are utilizing her patented innovations to harness the power of early language exposure to improve outcomes in babies, all babies, not just babies in the NICU, but also babies at home. Talk to babies. I love saying that because it's a really important concept to understand. Babies use infant-directed speech, the adults in their life speaking to them, as a sort of stimulus to wire their brains to process language. This is one of the biggest accomplishments in early childhood, and it's a really important part of early brain development. You see they're creating the neural connections to process speech sounds, and this is the first step in receptive language learning, and it's intertwined with brain development. In fact, the quantity, quality, and variety of language that infants are exposed to during the first months of their life are directly related to the outcomes that everybody cares about that you see here below. Now let's look at the first problem that we're going to solve using our technology. And think of this as sort of the roots of small talk. This is around prematurity or babies in the NICU. About 10% of babies don't go home with their parents when they're born. They stay in the neonatal intensive care unit, usually for clinical situations arising from prematurity. Now, we can all sort of recognize the emotional impact that leaving your baby behind has on families. But it's actually a bigger problem. This lack of exposure to infant-directed speech, these babies in the NICU get about 10% of that infant-directed speech than their peer babies who go home. This creates a brain development problem due to a lack of infant-directed speech. Using a grant from the NIH, Smalltalk has developed a solution. Our Connect speaker device safely brings recorded maternal or primary caretaker voice to the baby right into the incubator or into the open crib. Our app can be used at the hospital on an iPad or at home on a regular smartphone to record lullabies, songs, and stories speaking to your baby. The speaker device stores that content, and then during fitting into nursing flow, a single button push will play it at a safe volume and for a safe duration for that particular baby based on their gestational age and the location of the baby. This supports their growth and development and stability. It's really, really important, that maternal voice exposure, but it also creates a sense of connection for the parents at home who can't spend the amount of time with their baby that they wish to. Now, I've talked about the importance of brain development and language, so can we take it a step further? Can we actually improve outcomes in premature infants using recorded maternal speech? The answer to that is yes. Now, this claim here, improving speech sound processing in premature infants, that's something we've met with the FDA about, and we are currently in process of running a clinical study. This we expect to be available in 2026. What you see on the right is a speech therapist doing a therapy session using our prototype device. The difference from what I previously described, the voice recording app, the Connect speaker, that's still there, but now we have a sensor-equipped pacifier. So the baby's suck strength is being constantly measured by the speaker device, and the speaker will deliver 10 seconds of that maternal voice to the baby when a certain threshold is hit. That's adjusted. It's really adjusted constantly. It's designed to engage the baby in a sort of back and forth, asking for this content. This is something babies recognize this contingency very, very quickly. And when it's FDA cleared, it'll be the first intervention to improve brain development in premature infants. How do we know it works? So we do have 3 peer-reviewed studies published. Natalie's innovation started at Vanderbilt University, where she demonstrated that something called ERP, measurement of speech sound processing. So ERP is event-related potentials. This is a time-locked EEG process. That baby in the top there you can see is wearing a sort of net of sensors. And they're being exposed to speech sounds, and then we're measuring how their brain processes those speech sounds. Speech sound processing at discharge from the NICU predicts 1, 2, and 4 year speech, language, and neurodevelopmental outcomes very powerfully. So it's a really, really important metric to work against. Using our prototype device, 2 to 3 weeks of active learning, using the pacifier sensor in a therapy session for 20 minutes, significantly increases speech sound processing in premature infants. This is what attracted our grant, and we're very excited, as I mentioned, to bring this to market, hopefully in 2026 after we complete our pivotal clinical study and gain FDA clearance from the FDA. Now, NICU babies graduate and they go home. And I'm skipping ahead for a second here. Our business model for the NICU is based on a product as a service business model, where we provide all the speakers and iPads that they need to implement our system. It's a pretty modest contract, averaging about $5,000 per year. We're making this available in the second quarter of this year. And then with FDA clearance, we'll bring the Connect Active, same business model, higher price per contract. Now, those babies go home. There are babies at home. So the second problem that we solve, or the second opportunity that we have, is really to influence brain development through foreign language exposure. There's a lack of tools and resources for introducing foreign language to babies. Babies raised in a bilingual household have numerous lifetime advantages, like linguistically, cognitively, and socially. Parents understand this, and they rank language skills among the highest aspirations for their children. 50% of parents today seek opportunities to expose their child to foreign language. 80% would be likely to purchase a technology solution for use in the home. And for those who go looking for a solution, 0% find one, because there is no solution on the market for children less than the age of 2. So Smalltalk is the first to create a language learning system designed specifically for babies. Babies are really effortless. They're very eager language learners. Our products enable any parent anywhere to create a multilingual household. And this early introduction to language with Smalltalk stimulates cognitive development, fosters cultural awareness, sort of laying the groundwork for a true global citizen. Our products range across a spectrum of digital, lesson kits that come into the home, and optional hardware. Our free app is available right now. That offers 7 different languages, exclusive cultural lullabies, songs and stories recorded by real parents, native speakers of those languages, with their own babies. So that infant-directed speech, which can't be imitated, is actually part of our brand. We structure our content into lessons that you combine activities with your baby, so that kind of engaged learning process. Will have premium content available later this year to add on to our free content that's available now. In the second quarter, we'll be launching our small story lesson kits. This comes specifically from our digital customers, asking them where they want to go next. They're interested in premium content, but they really like the idea of books and learning kits coming to the home. Learning kits are structured around 2 books written by native speakers of that language, so real people. We bring companion audio, companion video, flashcards, activities, all for about $35 a kit, and we'll have ultimately 18 kits available, spanning all the way up to 24 months of age. Then our optional hardware is a speaker device designed specifically for the home environment for the baby, and a sensor-equipped pacifier, as you learned about before. This elevates the experience with our foreign language content and accelerates brain development as well. So our user journey is really, we make Smalltalk easily available to anybody, a casual user. Anybody who's been exposed to our brand in the NICU is part of our, they have an account with Smalltalk. They come into our system that way. Anybody who's casually interested in this can easily engage with our brand through this free app. If they're engaged, they can up to optional content. They can move into the lesson kits. And then lastly, our super users are the group that is more interested in the speaker device and the Passy device for home. But our goal ultimately is to create a global community around language learning for preschoolers at home. From a competitive environment, there's already 40 million families globally using technology in the home for language learning for their preschoolers. The big player here is Lingo Kids, which has over 25 million of those families. But all of these solutions are for ages 2 to 6. Only Smalltalk is focused on that 0 to 2 lane. This is what we think of as our beachhead, and then eventually we'll be expanding to compete directly with the entire category. Previous to 2021, we focused on publishing research, patents, prototypes, talking to our users. We developed our app, brought that to market in 2022 and 2023, validating a product market fit, getting a lot of input from our users. We're launching the lesson kits. We're launching the NICU Connect speaker. We'll be doing some early launch on the Passy and Egg late in 2024. 2025, moving to international markets, because globally, language learning is English language learning, and then eventually scaling into toddlerhood. We've published peer-reviewed studies. We have issued and pending patents covering our hardware technology and connecting sensor devices and speaker devices for language learning and brain development. We have our grant that I mentioned, and we are ready to launch into both ends of this market. From a team, my background, I've been in life science startups for about 25 years at Adiza Biomedical, which innovated the first diagnostic test to predict preterm birth. I founded and led the commercialization team, was vice president of marketing at our IPO, and then our ultimate exit for $450 million. From there, I co-founded Servalens, a device to predict preterm birth, and eventually sold that to Unified Women's Healthcare. I was a venture partner at Jumpstart Ventures, so I've done investing into the life science space as well. Natalie is a key opinion leader in brain development for infants, highly published, highly respected, does incredible work in her laboratory, and really leads our scientific approach to everything that we do. Jess is a highly experienced product manager, product developer, brand strategist, and really is our voice of the customer. And Annalisa came to us actually as a very first adopter of our app with her child, and she is a bilingual Italian and specializes in language learning for preschoolers. And so she really drives how we approach our consumer language learning products. I really, really appreciate the incredible support of Nationwide Children's Hospital, along with these other important investors and partners. I can be reached at dean@smalltalk.tech, and I would welcome any message or follow-up from any of you. Thank you for your time.
It's great to get applause when you didn't do anything, but I'll give it to Dean. But happy to answer any questions. Go ahead.
Sorry. Thank you. So just from a standpoint of looking at the capital strategy for the progression of this company, are they fundraising right now? And,
Yes.
how much have they raised to date in, dilutive funding, I guess?
I can't give you exact numbers, but I can tell you that the trajectory that they've taken through funding has been it's been heavily influenced by the fact that they got a Phase 2 SBIR grant. And so a lot of their funding right now is coming from that. I think they have done a seed round maybe on the order of like $2 to $3 million, but they are looking for additional capital. I know that they are actively fundraising right now. Great. Dean would be happy to follow up on that question, I'm sure. Any other questions? Yeah, right here.
Yeah, question I had was, what is their regulatory strategy for this? And I know they're doing a product as a service model, but is there an eventual play to get reimbursement codes?
That's what they're hoping for. So there are some strategies that they're using right now just for speech-language pathology and hoping to be able to use some of those existing CPT codes for that. But as always, reimbursement is a huge question for these types of companies, and getting reimbursed is a major inflection point for them. Ultimately, that's what their hope is. They are going through medical device approval to get some form function claims, as he mentioned. So they are hopeful. Cool. Well, thank you very much, everybody, and on to the next speaker.
Okay, good afternoon, everyone. My name is Ellen Zaluka, and I'm a licensing associate
Hi.
in the OTC. And I'm happy to introduce our next presenter, who is Dr. Alan Cadotto. Dr. Cadotto is a surgeon in the Department of Orthopedics and is also the director of the Center for Orthopedic Innovation. He will be presenting on the early success stories and the inner workings of the center.
Thank you, Ellen, and to the OTC, I very much appreciate the opportunity to present this afternoon. This talk is entitled Orthopedic Innovation at Nationwide Children's, Leveraging Our Resources, Expertise, Technology to Advance Orthopedic Care for Children. This presentation will include off-label use of devices and product trade names. The objectives of this talk are to describe the interconnected processes of surgical innovation, research, education, and commercialization, highlight the resources available here, and demonstrate the immense potential of interdisciplinary collaboration between academia and industry. I used the Google generative AI system to generate a definition for innovation. And it describes innovation as the process of creating new ideas, products, services, or methods that have a positive impact and value. There's a significant innovation gap in pediatric orthopedics. The FDA estimates that pediatric medical device development lags 5 to 10 years behind that of adults. There's a large amount of dollars that go into MSK healthcare, as we all know, but there's a very minimal amount of that goes into MSK as far as development. Most approved devices are for patients older than 17. More on the innovation gap. Surgeons are required to use off-label devices. As a result, we repurpose adult design devices which may have suboptimal design, fit, or material properties. This oftentimes presents a moral and ethical burden that surgeons must overcome. In addition, there's a regulatory lag. Pediatric devices, the definition is under the age of 21. A recent study looking at 124 Class 3 recently approved devices, only 2 of those were dedicated to pediatric use. We all know some barriers to innovation in pediatrics. Children have changing sizes and physiology. They do unique activities. There's a significant cost to bringing a novel device to market. And in general, it's been historically shown to be a smaller market and lower reimbursement. So ultimately, in our field, the burden falls to surgeons and other providers to continue to advocate for device development rather than make do with what we have. So in our field, it's our responsibility to offer the most efficacious and cost-effective treatments, be cognizant of the scientific basis for these treatments, be proficient in any new treatment as well as the labeled indications, safety, and efficacy data, and offer appropriate training methods for these new technologies. There's a lot of good news around this as well. I presented some bad news, but the good news is you've already heard. So we have this Nationwide Innovation Center that's developing with the help of Dr. Corey Chris. The humanitarian device exemption FDA regulatory pathway has been very instrumental in approving important implants and instruments that we use daily in our practice. And then the system of hospitals for innovation in pediatric medical devices. So looking around this room, looking around this hospital campus, you can really tell that there's a lot of resources, interest, and expertise, and really the environment and ecosystem in Columbus in general is a very special one for innovation. Around every strategic plan, innovation is central. So what are our goals in this endeavor? Number 1, to improve landscape for pediatric orthopedic innovation, foster collaboration between healthcare and industry, streamline regulatory pathways, and increase market compensation to further drive progress. The ultimate goal being to help children. So we set out about a year and a half ago to start a effort that we coined the Orthopedic Innovation Center. And we partnered with the Office of Technology Commercialization with the mission to leverage our resources here, expertise, and technology to advance orthopedic care. We constructed a team with several members in this room from OTC and the Department of Orthopedics. And if Matthew hadn't already given you a standing round of applause, I would have done so now. They've truly been instrumental in helping us in this effort. So where are we now? Again, we started about a year and a half. We have a monthly ortho innovation review with our stakeholders. And our productivity metrics are listed below. But really, with the help of the OTC and the resources available to us, this has really been a crawl, walk, fly phenomenon. So I'd like to go forward and give you some samples of some of the projects we're working on, just to get an idea of what we're able to achieve and some of the things that we can do with the resources available to us. First project is this new model for scoliosis, a biomechanical model for simulation, training, and research. Scoliosis is a huge passion of mine. Spinal deformity, pediatric spinal deformity, it's a big component of orthopedic practice. Scoliosis is a 3-dimensional deformity, but currently available models do not allow us to simulate and train appropriately for our trainees and for ourselves. There's also a lack of biomechanical testing and analysis, and a lot of this ends up being expert surgeon inputs and biomechanical studies that do not translate. What is currently available as far as a model? So we have low fidelity models on the left. We use these very often in training sessions. You might see these at industry meetings or national meetings where they're trying to display implants or train surgeons. There's also high fidelity models, but these are limited in their segments and they're not curved. Scoliosis is a deformity. So the solution is a novel 3D scoliosis model with optimized biomechanical and viscoelastic properties that mimic real-life scoliosis correction. We wanted to mimic the real bone screw interface, spinal stiffness, strength of the ligaments, tendons, and discs. We also wanted to have the same resistance to correction, the stress that our screws and rods and implants experience during the actual surgery correction, and appropriately simulate these forces needed in surgery. This is an example of our first prototype, 3D printed based on patient MRI and CT data. Again, simulating the bone, discs, ligaments, tendons. This is prototype number 2, informed by our experience with prototype number 1. Again, trying to create this viscoelastic environment for scoliosis correction. And one of the best parts of this is working with your partners and colleagues in the hospital to figure out the best ways to go forward with the project. Future directions of this project are to establish a standard for education and proctored simulation prior to the real surgery. The vision here is to get the model, put it into a skin, take it to the lab, train some fellows and residents, but also use it for research. And this will also inform future fusionless spinal technologies. In the world of spinal deformity, spinal fusion is the biggest operation done now. There's a huge push for fusionless surgery, and unfortunately poorly informed at this point. The second project I'd like to demonstrate is one of brace compliance or brace adherence. In orthopedics, we treat a lot of scoliosis, club foot, and other diagnoses that require brace compliance to promote outcomes, good outcomes. And we know that the brace is dose dependent, or the outcomes based off of this are dose dependent. Currently available brace adherence systems will generate a report card. These are provider-centered or provider-facing. There are also some patient-facing platforms that have the sensor in the brace and provide a report card. The goal of this project is to create an all-inclusive platform that has bidirectional communication between the patient and physician, and also has a cloud platform that generates some kind of behavioral analytics, gamification. Think of your smartwatch on your wrist and the feedback that it gives you if you haven't exercised for a week. We know that outcomes are tied to brace adherence, and we can truly achieve really good results with bracing. The picture on the left is an example of the sensor within the brace that is commonly used, and this is the girl that was depicted in the slide before. This is our team of inventors. Dr. Tulchin Francis is our Director of Orthopedic Research. She has a lot of experience in these brace software platforms. Dr. Heather Richard, our orthopedic psychologist, she's instrumental in designing the behavioral analytics behind this. And then Holly is our nurse practitioner on our team who runs our brace clinic. And we're partnering with industry as well to hopefully get this to the next level. Next project is one that I think hits home for many of us, particularly the physicians working at a tertiary referral center such as this. There are a lot of medical conditions that we may not have the appropriate tools to handle, and this is common in our pediatric world, unfortunately. This is an x-ray of a patient who has a severe spinal deformity, is severely medically complex, is a poor candidate for other treatment options, including bracing, casting, or posterior-based instrumentation, which is all we currently have, and has prior failed operations. The goal for this child is to optimize pulmonary function and slow progression of the chest and spine deformity. So with that being said, we wanted to put this child in something called halo gravity traction, but a program does not yet exist to do long-term halo gravity traction for a child like this who is medically unfit to undergo surgery. So we worked internally in our team to develop a program utilizing this process, or I would call this the stage 1 process. Number 1, we attempted to coat the pins for the halo device with hydroxyapatite to get better bone fixation in the cranium. Worked with partners in town to find patient-specific solutions for the wheelchairs, walkers, and other equipment. We developed patient education material, including the dynamic traction setup instructions, car transport plan, medical device information, and discharge logistics. And we worked very closely with the business process team, who also helped us get in touch with legal and the leadership here. Over a year ago, we discharged our first long-term halo gravity traction patient at Nationwide Children's. She is still in traction. This is her depicted in the clinic. Picture of another child, again with a severe spinal deformity that we enrolled in our long-term halo gravity traction program. Again, body habitus that is not fit for posterior instrumentation, severe spinal deformity, and we just want to optimize the pulmonary function of these children. Another project we've been working on is with Battelle. They came and gave us a presentation over a year ago, and we saw a lot of potential in their technology, their NeuroLife sleeve, for our pediatric population. Their NeuroLife sleeve was developed with the premise of the adult population, but given what we know about pediatric neurophysiology, this has a lot of potential. And without giving too much, we will be starting our first clinical trial with them in the summer, likely. It's important when you talk about innovation to acknowledge novel applications of old technologies. Tomosynthesis is an old technology for imaging, but this is a new application of software to this old technology that we used here and we trialed here, and we were able to mold the feedback given for this. So this it's a good example of using that old technology, but then being able to stitch the radiographs, navigate the spine, and using AI to segment the spinal levels during surgery. Also, an old technology is descending neurogenic evoked potentials. This is a technology called dynamic spinal cord mapping, utilizing the old system in a new way to localize a lesion during spinal deformity surgery if you have a neuromonitoring alert. We have very talented folks in our Department of Orthopedics, and I will say innovation happens regardless of having a center. And we have fierce innovators in our department. That was the surgeon team, our APP team. You know, if you look out the window of this place, you'll see the Livingston Orthopedic and Surgery Center. We are very proud to be moving into this in 2024. But it will house our new gait lab, our new spine motion analysis lab. Again, both of those things being integral in a lot of the innovation projects we're working on. Those are some renderings there, but again, just take a peek outside. Dr. Telchin Francis is our Director of Orthopedic Research, and with her help, we are involved in multiple national and international study groups. She also runs our Honda Center for Gait Analysis and Mobility Enhancement. This new spine motion analysis lab will be one of the first in the country, which we are very excited to pursue. The idea being, with motion-preserving technology, how does that compare to our current gold standard of fusion technology? And then will that further inform how we do spine deformity surgery? If you're interested in golf or ballet, you can enroll yourself to be a control in this cohort, or even bring your children in. It's kind of a fun day in the lab. Our group is very involved in education and training, and as we think about innovating our education process, how do we get from a intern to a very competent surgeon safely in the modern day? We are starting our own Nationwide Children's Hospital Pediatric Orthopedic Innovation Fellowship. And the idea here is to take a pediatric orthopedic surgeon who is interested in learning the process of innovation, regulatory pathways, licensing, and bringing that into an actual training program. This will be the first of its kind in orthopedics that we know of, or at least pediatric orthopedics. In the Department of Orthopedics, we represent all major subspecialties very well. And in conclusion, there's innovation everywhere. You don't necessarily need a center. There's fierce innovators all around. But if you look around this hospital and in this room, the environment is ripe. We have the highest level of clinical care, academia, industry, investment, tech transfer, and expertise. And we asked, why not? Can a dedicated infrastructure propel us to the next level? Thank you.
Thanks for the presentation. Quick question. So do you have an in-house team to develop this device, or do you outsource?
Which device?
The device you just mentioned, like those devices, do you develop those in-house, or do you outsource?
Yeah, I think, one of the things that we really appreciate is that we have a lot of resources in-house. Obviously, there are some resources we don't have. And this is where I think partnerships are really important for us. A lot of these projects we are partnering with external groups to help us, but some of the stuff we're doing in-house. So I guess it just depends on which project in particular.
Yeah, I was for the simulation for scoliosis. That seems like a nice market, a great market that can be expanded to, like you said, getting rid of fusion on the spine as well. In terms of the software itself, just maybe a few more seconds or a few more words on some of the uniqueness and robustness and different things with the simulation software around that.
Yeah, absolutely. I would say it's very much far away from a software at this point for the simulation for the scoliosis correction. Is that the project you're? Yeah. It's really truly a 3D model at this point, and we're using a lot of feel for scoliosis correction, deformation of the rod during correction, and we're using techniques like osteotomies, facetectomies, resection of the ligaments to figure out, is this truly a viscoelastic model that is going to be good for us simulation-wise. Once we arrive at the appropriate formula, we're going to translate this to varieties of spinal stiffness, and that's where we're probably going to do biomechanical studies to measure the stiffness of the spine. And then, using our flexibility films, we will be able to extrapolate patient-specific, how stiff do we want that model to be for that patient. So in theory, pie in the sky, is we will have several models for scoliosis with different curve types along different stiffnesses, along different magnitudes of severity, so that you can potentially choose one and either buy it or simulate it. The step after that, because it's a 3D printed model from MRI and CT from patients, you can actually make a patient-specific model for my case coming up on Tuesday and simulate that with your trainee. So this is our thought process behind it, and I think there are some softwares we are working with now that can simulate the stiffness of the spine using a certain algorithm from the standing X-rays and flexibility X-rays. And then how do you throw the spine motion analysis into this will be a completely new endeavor.
We do have an online question from Cecilia who says, this isn't my area, but is there any insight into molecular causes of the spinal problems in children?
Yeah, this is a huge area in our national and international societies. There's a lot of work being done on genetic research for scoliosis, and then from a molecular standpoint, using one of the collagen models in the blood at the time of scoliosis progression. So I would say very briefly, there is some work being done at a molecular level and a genetic level to understand the etiology, risk of progression of scoliosis. We've had some internal discussions on doing some discussion on can we affect the flexibility of scoliosis with a molecular model. This is a very limited space in orthopedics. Orthopedic surgeons think of us as screwdriver and hammer people, but I think partnering with the right people, we can learn a lot more going forward. Thank you.
Thank you very much.
Hello, my name is Jocelyn Idol. I'm one of the senior licensing associates, and I'd like to introduce our next presenter, Dr. Paul Martin. He is a principal investigator in the Center for Gene Therapy and also a professor of pediatrics, physiology, and cell biology at The Ohio State University. Today he's going to give a very nice overview of a new startup company that we're forming here called Genacera. The company is utilizing a lot of IP coming from his NCH lab. So now I'll hand it over to Paul.
Thank you so much, Jocelyn. So it's my great pleasure to have the opportunity today to introduce you to Genacera, which is a new startup company that I co-founded, whose goal is to help develop and bring to market some of the next generation gene therapies that have been developed in my lab to treat muscle diseases and lysosomal storage disorders. We're headquartered on Kinnear Road in the Rev1 innovation space, and we're very happy to be clients and advisees of Rev1 Ventures. So I co-founded this company with Guidon Ackler who's a physician and medical geneticist who specializes in the genetics of gene mutations that affect the Iranian Jewish community. And our lead disease that we're studying is, in fact, a disease that is very prevalent in this community. We've been joined by some excellent business partners and leaders in Patricia Beckman, who's our CEO, and Ron Medley, who is our Director of Business Strategy and Development. So the problem we're trying to solve is a very not insignificant problem, which is that gene therapies are not universally effective. And what I mean by that is that the simple idea of gene replacement, of putting a correct gene back into a patient where there's a mutated gene that's lost function, will at best, almost always, just stop or slow the disease process. So if you're treating a child very early in the disease process, that can be very effective. But if that child has significant disease already, and for the muscular dystrophy, certainly these are debilitating diseases, the best case scenario goes to lower and lower levels of efficacy as the disease progresses, because you really are not reversing the disease, you're simply stopping the disease from going further. So our goal is to create new gene therapies where there are multiple functions, or multiple genes in this case, within the same AAV vector, where they add the ability not only to stop a disease from happening through gene correction, but also to reverse pre-existing disease symptoms. And for muscle diseases, this looks something like this, where we have two genes now instead of one that are packaged into the same AAV vector, one providing gene replacement to prevent further disease, and one providing a muscle booster gene to build new muscle mass and strength. And so these muscle booster genes, there are a number of them, but we've happened on one that I'll talk to you about, really do three things. One, they stimulate muscle stem cells to activate and regenerate muscle. Two, they can build on the size of pre-existing muscles so that those muscle cells become bigger, and then the bigger muscle cells do more work. And the third thing is they can build new muscle cells and tissue altogether. And that is also an incredibly important property to reverse muscular dystrophies. They can also, these strategies can also be applied to diseases that are not genetic. I don't know if you call it a disease, but it's aging. So sarcopenia, or the natural loss of muscle mass during aging, is another application which we think will be particularly opportune for this strategy. So the company is in the process of licensing 6 pending patents from Children's to develop a strategy for this dual gene vector approach, as well as applications directly targeting 3 particular diseases that I'll tell you about. In addition to those 3 particular diseases, though, 2 of those therapies have secondary markets in processes that affect, instead of thousands of patients or hundreds of patients, as rare genetic diseases do, potentially millions of patients. So GNE myopathy is a progressive adult-onset recessive genetic disorder that results from the loss of the GNE gene. So the GNE gene encodes an enzyme, a dual enzyme protein that is the committed step in sialic acid biosynthesis. So sialic acid is a sugar that's at the end of many glycan chains on proteins and lipids throughout the body. There are no FDA-approved therapies for this disease. A glycan replacement therapy has been tried all the way up through phase 3 clinical trials, and none of the clinical milestones were met in those trials. There are at least 40,000 patients worldwide. That's based on what are the known mutations currently. Obviously, it could be more as more disease mutations are identified. And in the Iranian Jewish population, the carrier rate is as high as 1 in 8 in different parts of the United States. So it's a disease that really has a profound impact on this community in particular. Our second application is for limb-girdle muscular dystrophy type R9. This is an FKRP deficiency. There are thousands of patients in the U.S. alone, probably 10,000 patients in the U.S. and Europe with this disease. There are no treatments that are FDA approved at this point, although single gene replacement is currently being investigated by several companies. So here again, we want to provide a dual gene therapy approach where we have the potential to reverse disease in addition to just stopping the disease process from moving forward. And then the third disease is lysosomal acid lipase deficiency. This is a lysosomal storage disorder that results from mutations in the LIPA gene or lipase A gene. This is the only enzyme in the lysosome that breaks down triglycerides into free fatty acids and cholesterol esters into cholesterol. So what happens in this disease is there's an enormous buildup of fats in tissues, which is akin to what one sees with obesity. There are two components to this disease. There's complete loss of function, which causes Wolman disease, which is a disease of infancy, where children don't live past 4 months of age without treatment. And there's a partial loss of function version of this disease called cholesterol ester storage disease, where people might be diagnosed as teenagers or young adults. This disease also can manifest as non-alcoholic fatty liver disease or non-alcoholic steatohepatitis. These are diseases that millions of people in the United States have as a result of obesity, and reduced LIPA gene function is a biomarker in those patients, and we believe that this gene therapy might be appropriate for treatment of these patients as well. So I'm just going to give you a few pieces of data. I know this might be asking a bit much, but this is a complex slide, but it really summarizes the entire approach and what we're all about as a company and as a lab. So one of the most profound functional tests in muscular dystrophy is walking. I mean, when children lose the ability to walk, that's a major milestone in the disease that impacts their quality of life. We do a lot of tests in the physical therapy department here to look at that, including what we call a 6-minute walk test. Well, mice walk much better than people, so we actually do a 1-hour walk test in mice, which is 3,600 seconds. And if you look at this little bar graph on the left, that's what wild-type mice do at 7 months of age, and 100% of mice can walk for the full hour. They just love to walk, and it's not a problem. In the second bar, you'll see what happens when we look at a mouse where we've introduced a human mutation that causes limb-girdle muscular dystrophy, the P448L mutation. Those mice lose about 2/3 of their ability to walk by the 7 months of age. And then we tried a single gene replacement, which is what FKRP is, at a low dose and a high dose. That's what LD and HD are. And what you can see is that you can improve the outcome measure with high-dose single-gene replacement, but you cannot reverse it entirely. And that's because when you start treating the disease after it's already occurring, you can only go so far. And this is the best-case scenario here. We're treating early in the disease and then asking for an outcome in the middle of the disease. As you go further and further along in the disease, single-gene replacement gets worse and worse at recovering this milestone. Follistatin is a muscle booster gene, FST, and we tried that alone too, and that really does nothing to improve walking by itself, which was surprising to us because it actually does build muscle mass, but what it does is it fails to build muscle mass evenly across the body plan unless gene correction is also provided. So what happens is you see the growth of some muscles but not others, and in the limb that leads to imbalances that really don't improve ambulation at all. And only when you put the two genes together in a coordinated package can you get walking back. And this is what we want, is this last bar where we have treated mice where 100% of them are walking just as a wild-type mouse does. So that's really the goal. And that's what we're all about at Genus Zero. I'll just show you another example. This is from the G&E myopathy work, where we just injected one or two of these lead compounds that are bicistronic dual gene vectors into a muscle, either the tibialis anterior, the TA here, or the gastrocnemius. And within 2 months of introduction of that dual vector, we can double the size of those muscles. And you can measure their strength as being doubled as well with physiological measures. If we engineer a dual gene vector with an internal ribosomal entry site or an iris, it works far less well than if we use a tandem promoter approach, because the genes aren't amplified in their expression, and so you need a much higher dose to get it to work. And if you just use the single G&E gene, which is the gene replacement gene, for either one of those two leads, they don't do anything by themselves. So not only can we get improved muscle function with this approach, but we can actually get it pretty fast. So one example from the lysosomal storage disorder work. So we use LIPA-deficient mice to do this work, which is LIPA minus here. And what you see on the top panel is a liver, and on the bottom panel is a spleen. And what you can see by 6 months of age is that the spleen has grown, the liver has grown in size to 4 or 5 times its normal weight. And you see this in patients too. Their abdomen becomes distended because their liver is so enlarged and laden with fat. If we treat at 1 to 2 days of age, so a preventative therapy, we can completely inhibit that disease from happening. And if we treat at 8 weeks of age, when the liver is already twice its normal size and already has highly elevated levels of fat, we can also reverse that disease back to normal levels. Now, we've engineered this gene therapy in such a way that it actually is a dual function therapy as well, even though it's a single gene. So it's a gene therapy that's only expressed in the liver, but it's engineered in such a way that the protein then is secreted out of the liver and can be resorbed by cells throughout the body through the mannose-6-phosphate receptor, targeted back to the lysosome, and provide therapy in trans to cells throughout the body. So it's really an enzyme replacement therapy generated by a gene therapy. So our platform of dual gene therapies is our goal, obviously, is to be the first company to bring these to market and test them to see how they fare against what the current gold standard is. We have plenty of proof of concept data to suggest that this should be successful, both in early and in progressively and severe disease. The technology can actually be applied to 23 different forms of muscular dystrophy. The gene size, of course, is the rate-limiting factor, but there are all kinds of tools one can bring to bear to engineer these for different disease forms. And with our approach, we hope to be able to treat non-genetic diseases such as muscle wasting in the elderly as well. Obviously, coming out of Children's, which has many INDs for gene therapy vectors and 2 FDA-approved therapies, we have an enormous amount of experience in how to move this process forward on all fronts, from manufacturing to clinical trials. And we have an amazing regulatory team here to support FDA filings. So I'd just like to thank my lab and our collaborators at both the Children's and at University of Indiana for their help with this work, and very much thank the Office of Technology Commercialization, who funded several of these projects, as well as several patient groups. So I'd be happy to answer any questions you have, and I'll also be at the reception this afternoon, and be happy to talk to you there as well. So thanks.
For these dual gene therapies, do these require dual AAV vectors? And how often is it necessary for both vectors to transduce the same cell for it to be effective?
So the question was, do you need to mix 2 AAVs together to do this? And the answer is no. These are engineered into a single AAV vector. And in doing so, we actually amplify the gene expression for both genes relative to mixing them as single vectors by about tenfold because of the regulatory elements we put in. The answer is no, and they would express in all of the same cells as long as the AAV goes into them. That is a major advantage. We thought about the idea of doing what you are asking about, and others have done these sort of mix and match experiments, particularly with follistatin and microdystrophin, and shown some benefits. But you run into a huge translational issue, which is you're going to now double the amount of AAV you ask to give to the patient, and in our case, maybe even quadruple or make it eightfold higher because we're getting amplified gene expression at the same time. So that is not an easy ask to do, yeah. All right, well, I don't see any more questions, so, oh, sorry.
Excellent talk. One question, you mentioned using iris elements in biosistronic expression cassettes. How do you overcome the difference in expression from your first gene versus your second? And are you able to modulate which gene comes first? Because typically, with my experience with iris elements, is that second gene is a lower or more unpredictable expression level. How do you compensate for that?
Yeah, so I showed that as an example of a negative result. What we do to overcome that is we use a separate promoter for both genes and a separate poly-A for both genes. And really, we spent a lot of time working on irises and inteins and other things that would allow a single promoter to express both proteins. And what we came up with time and time again was that it was just very disappointing. So what we decided to do is just do the simplest thing you can do, which is just to make each gene a separate RNA driven by a separate promoter. And what we found is that by doing that and having enhancers in those promoters, we actually amplified expression for both genes when the right combinations were put together. And that led to much more potent therapy and more controlled.
Dr. Martin, we have a virtual question from Gabe. Are there limitations to the genes that can be incorporated into the dual AAV technology because of gene length?
Absolutely, there are limitations to the size that you can use in our lab, we like to say 4.8 kilobases is the packaging limit of AAV. People will say 4.7. Certainly there are vectors that have had 5 kb packed in. You tend to lose yield as you go up. So even if it's packaged, if you're manufacturing it, it can lead to a lower yield as you add on past that magic 4.7 number. So for the disease we targeted, the 2 diseases, the target genes were 1.5 or 2.2 kb, and then adding on follistatin, which is just a kb, in addition, allows you to do a lot with the regulatory elements. And as you get to bigger genes, what you can do becomes more limited, and then eventually becomes impossible. Yeah.
Okay, thank you very much,
Good,
Dr.
thanks.
Martin.
Hello again, everybody. It is my pleasure to introduce our next speaker, Dr. Aaron Trask, out of our Center for Cardiovascular Research. We'll be presenting research that he's done and a novel algorithm for gathering insights from the fluid dynamics of the microvasculature.
Good afternoon, everyone. I want to start by thanking our Office of Tech Transfer for the opportunity to present our work. So I'm going to tell you a story about how I'm a cardiovascular physiologist, pharmacologist by training, and I will tell you how I stumbled into machine learning today. So I'm going to try to convince you over the next several minutes that this statement is true, that blood flow patterns harbor more useful information than we currently utilize. And my lab is primarily interested in coronary microvascular disease, in type 2 diabetes and metabolic syndrome, conditions like obesity and hypertension, all of which have an element of coronary microvascular disease. And so what you see here is, let's see if I can get a pointer. Here we go. What you see is a coronary blood flow pattern obtained by Doppler echocardiography in a mouse. And so we can measure this very reasonably in a mouse. And you can see that the blood flow pattern actually is very unique in its shape. Currently in the clinic, we only use the peak of that blood flow pattern as an index of microvascular health and blood flow into the heart. The second part that I'll tell you about very shortly is how we've essentially taken the lessons in the coronary system to the umbilical system, mostly as a way to predict the development of fetal growth restriction during pregnancy. And in that condition, my clinical colleagues, Mark Santillan at the University of Iowa, will use things like the systolic peak and this diastolic trough and the ratio of those two to sort of assess and follow a pregnancy as it progresses, if it's indicated or at risk for fetal growth restriction. So we'll start off by talking about coronary microvascular disease. Many of you have probably seen pictures of the heart like this. This is the heart. These are the coronary microvessels that come off. Most people think of coronary artery disease as large artery disease where you get atherosclerotic plaques that eventually lead to flow limitations and heart attacks. We're interested in the microvessels that are downstream of those. These are the tiny blood vessels. If you look at your hair, they're the diameter of your hair. They're the ones that regulate blood flow and the ones that my lab is interested in. And as I mentioned, coronary microvascular disease underlies diabetes, high blood pressure, obesity, metabolic syndrome, and other conditions. So if you were to put the timing of coronary microvascular disease as a function of the timing of diabetes or metabolic syndrome, you get a progressive decrease in blood flow over time. And the initial sort of factor that happens is this microvascular disease. And other studies show that, so other groups other than us have shown that there's a functional deficit, so they don't dilate as well. They have more tone, but we've shown in our past work that these blood vessels actually remodel. So they're structurally limited to have a smaller lumen, and the smaller lumen or opening for blood flow means that there's a reduction in blood flow. That's followed shortly thereafter by macrovascular disease, so stiffening of the large arteries in the coronaries and other peripheral blood vessels, and ultimately culminates in what we all know as coronary artery disease, which is shown here, which is evidenced by the presence of atherosclerotic plaques, which reduce the lumen for blood flow, ultimately leading to a heart attack. And so my lab is very interested in this early process to see if we can prevent the progression of this entire process early. The thing that I always like to point out to people is that most of this process is subclinical or asymptomatic. Many of the patients who have this don't know that they have it. It's not until you get to 80 to 90% plus blockage of your coronary arteries that there may be symptoms. And so we were trying to get at basic mechanisms of understanding how the coronary microcirculation interacts with the surrounding myocardium, and that's sort of illustrated here. So this is a coronary blood flow pattern just with our own defined phases of that pattern. And you can see when the heart's contracted, there's not a lot of blood flow. So when the heart's contracted, it's essentially squeezing those blood vessels shut. And it's only when the heart relaxes in this diastolic phase that you get the most of the coronary blood flow. And as I pointed out on the first slide, you can tell that that has a very unique pattern. And so we were doing a series of experiments in our lab and blood vessels to try to understand the basic mechanisms of that. And we were never satisfied with the data. And so Ian Sunyes was a very talented biomedical engineering undergraduate student in my lab at the time. And I said, Ian, let's just see if we can do some slopes and sort of define these phases of the flow pattern, because we know that this is a representation of the interactions of the coronary microcirculation with the surrounding heart, and see if those correlate to any of our microvascular data. And so this is the table that he showed me several years ago now. I don't want you to go over, I don't want you to pay attention to the details, but understand that the flow pattern properties that I just showed you were correlated in many ways to the coronary microvascular properties that we talked about, so remodeling mechanics and things like that. And that's useful from a basic science standpoint, from our laboratory standpoint. But the thing that really stood out to me was what this column over here on the right is that some of these correlations were unique to only normal mice, and some of these correlations were unique to diabetic mice. And that got me to thinking about how coronary microvascular disease is diagnosed. So currently, there's no direct way to measure or diagnose coronary microvascular disease. It's all done indirectly. So if you come in with chest pain, but your coronary arteries are clean, so there's no evidence of atherosclerosis, then a physician may diagnose you with coronary microvascular disease, but there's no guarantee. And so we asked the very simple question, can we use these blood flow patterns to predict coronary microvascular disease? Again, this is another representation of coronary blood flow in a mouse. And so, as I told you, Ian was a biomedical engineering by training, and he had a lot of skills in MATLAB, so it's a bread and butter software program that's used in the engineering field. And we took the correlations, so these sort of slopes and point velocities that we had, and he built an initial predictive algorithm using MATLAB. And in 15 mice, this was the very first experiment we ever did, he was 75% accurate. I was floored. I was like, that's 50% better than chance, so I think we're onto something. And he presented this at an internal research retreat at Nationwide Children's several years ago, and Chris Bartlett came up to his poster and talked to him about the potential of using machine learning on the blood flow patterns to sort of increase our accuracy, because machine learning is able to pick up subtleties that maybe you and I are not able to pick up. And we brought in Will Ray, who is an image processing expert. And at that time, we wrote a grant to the NIH on an R21 Trailblazer that was thankfully funded to essentially test whether or not we could do machine learning to pick up these subtle curvatures and other things in the image that maybe we can't pick up and do machine learning. And we did this in 500 to 600 animals, and I'm going to tell you folks that that was a heroic effort by the people in my laboratory to do combined coronary Doppler echocardiograms, invasive cardiac catheterization, and echo all in the same mouse around the same time. So we call it semi-simultaneous. And where we're at right now is currently the images alone are 80% accurate. As I told you, 80% accurate at identifying a coronary microvascular disease animal versus a non-coronary microvascular disease animal. And if we add in things that are clinically relevant, like cardiac function, so if someone goes in for a cardiac workup, they're going to get an echocardiogram. So if we add in these types of data, we get to 92 to 96%. So this is still an ongoing a process for us right now. We're still sort of fine-tuning these data, but this is where we stand right now at 92 to 96% with the addition of cardiac function. And so I was at a conference several years ago, and I was talking to my good friend Mark Centillion at the University of Iowa. He's a maternal fetal medicine doc, OBGYN. We don't do anything in the same field. We were just friends, and he said, "What have you been working on?" And I just told him what I told you. And he said, "We should do that in pregnancy." And I was like, "I don't want to work in pregnancy." But he converted me. And so he explained to me that during pregnancy, when a mother goes in for her standard 20-week ultrasound, in some cases, the fetus is measured small for gestational age. And if that's the case, then clinicians like Mark will order a Doppler ultrasound of the umbilical blood flow pattern. And they will use the systolic to diastolic ratio as sort of an index to track whether or not that pregnancy is at risk for developing fetal growth restriction. So obviously, you don't want blood flow restrictions to a growing fetus. And so this is a large problem for his practice and other practices like that. And so we essentially hypothesized whether or not we could take those images, so those Doppler umbilical cord or umbilical flow images, and subject them to a similar machine learning protocol that we did in the coronaries to discern whether that pregnancy would progress down a normal path as what we would hope, or if that pregnancy is at risk for fetal growth restriction. And this is sort of the first experiment we did in a subset of 107 patients from the University of Iowa. So Mark and his wife Donna have a huge database of 24,000 patients that we can use to test this on. So it's very ripe for this type of work. So this was the very first experiment we did in 107 patients. So first we started off with a simple logistic model of just the systolic to diastolic ratio. And when we started this project, Mark told me, he said, you know, the systolic to diastolic ratio has some prognostic value, right? It's useful in some ways, but it's not foolproof, okay? And so when we did the logistic model, we were about 70% accurate. And that's kind of in line with what Mark had indicated anecdotally, is that it has some prognostic value, but it's not foolproof. We then did machine learning on that ratio in and of itself, just to sort of see if the systolic to diastolic ratio had any value with a machine learning standpoint. And again, it was 70% accurate. So it's like this machine learning or systolic to diastolic ratio is kind of limited, I think, at 70%. And then we added in clinical variables, so maternal status, body weight, blood pressure, BMI, and things like that, and it increased to 77%. And then we just took the images, pre-processed them like we did in the coronaries, and just did machine learning on the images alone. And we were floored that it was 93% accurate in accurately predicting the development of fetal growth restricted pregnancies. And so our goal for this is not only to sort of further develop this in a larger data set, right, in order to predict the development accurately of fetal growth restricted pregnancies. But the other thing that we're interested in would be useful from a clinical standpoint is if we were able to sort of predict days from diagnosis to delivery, right? And that's something that we're working on because the difference between life and death for a fetus under Mark's care can be 1 day. And so if he had some reasonable way to predict using a very large data set like this, from days from diagnosis to delivery, it helps him better manage that and better manage pregnancies overall. So to summarize, current studies are underway to further develop, test, and validate both of these data sets. Machine learning of blood flow images and patterns may be a novel way to learn about and better diagnose diseases such as coronary microvascular disease and fetal growth restriction. Our vision for this is that this type of software algorithm would be put onto ultrasound machines. These are some of the major players in the ultrasound field, or to make the algorithms and software available on a web-based fee-for-service platform. I don't do any of this alone. I told you I was a cardiovascular physiologist, pharmacologist. This is the team that in my lab that has done a lot of this work, Patty McAllen-Hart, Yuki Oyama, who I told you did the heroic effort of doing all those coronary blood flows and cardiac functional studies in 500 to 600 mice. Jamie did a lot of the image processing, and Sam's worked on it. Ian and I started this project, I think 2017-ish, so quite a while ago to get this off the ground. Keishan and Michael both have worked on it. And then these are the collaborators that we've sort of made this team out of. Chris Bartlett in the Institute for Genomic Medicine does all the machine learning work. Will Ray does the image processing. And it really takes all of us in order to make this type of work possible, and I'm fortunate to be able to work with these folks. And then Mark and Donna Santillan and Olivia Peters at the University of Iowa, they really have a rich data set, and they're fantastic collaborators. We've been funded by the National Institutes of Health and seed funding from Nationwide Children's and the University of Iowa. And with that, I will be happy to answer any questions.
Dr. Trask, it was an excellent presentation. So when you're thinking about the monitoring, there are limitations with ultrasound. So would you have an advantage if you were able to do low-cost continuous monitoring?
In terms of like telemetry?
For fetal monitoring. So if you were able to do continuous monitoring using low-cost, non-ultrasound system, would that give you an advantage in terms of being able to monitor the fetus, the mother, and?
Potentially. We haven't really discussed that as a group. I think the benefit in our mind for the fetal project is that, if the algorithm comes back and says that a pregnancy is 99% likely to progress to fetal growth restriction, then Mark's going to monitor it more closely because it's at high risk. The flip side to that is that if it comes back and says that this pregnancy is at a low risk, then there's less frequent monitoring needed, and so it's less office visits needed for both the patient, but it's also less burden on the medical system. And so I think that there's a double-edged sword. I don't know if that sort of answered your. Okay, I'll be at the reception after. Thank you for your question.
My name is Kyle Murrah. I'm a senior licensing associate. I have privilege of introducing our next speaker, Dr. Tendi Chang. He is an otolaryngologist, and he's also part of the Center for Regenerative Medicine. And today he'll be talking about a device for tissue engineering of an airway device.
Thank you, Kyle, and thank you to the Office of Tech Commercialization for allowing me to present our work. I have no disclosures. I'm a pediatric otolaryngologist, and for those of you who don't know, I'm a surgeon that deals with problems basically from the collarbones up, excluding the eyeballs and the brain. But oftentimes we are left to cure disease of the head and neck region, and the resultant reconstruction really leaves us asking for what the ideal tissue source is. There can be significant functional implications and cosmetic implications whenever you perform surgery in the head and neck area, and this is something that was recognized even 400 years ago. This is a medical textbook where they described the need to remove a lesion from a gentleman's nose, and in order for them to correct it, they basically took a little island of skin and then they sewed it to his nose, and then he was left in this uncomfortable position for a period of time to allow for that island of skin to engraft. And so this has definitely evolved, where treatment of skin cancer can be reconstructed by borrowing tissue from the forehead, called a paramedian forehead flap, and ideally reconstruct cosmetic results. So these issues are abound in the head and neck area, and for me, we take care of a lot of children that have airway stenosis. And airway stenosis arises from intubation most commonly. Following COVID-19, there was a 40% increase of laryngotracheal stenosis in the adult population because of the need for mechanical ventilation and prolonged intubation. And in those situations, the workhorse for reconstructing a trachea or a larynx is also a tissue source. And there's no great off-the-shelf solution for these types of problems. And so what we're left doing when we need to reconstruct the airway is we borrow from the rib cage. So this is an approach that you see here in this image, where a child is getting a piece of rib that's harvested, and it's fashioned into the shape of a graft. The airway that's narrow is then expanded, and that rib graft can then be inserted to expand the airway, allow them to breathe better. But even there are limits to this type of surgery. You know, there are children that are born without a windpipe, or that are born without a voice box, and this is actually one of the first patients that I took care of when I began my career as a pediatric otolaryngologist, and that they were fortunate, quote unquote, to be resuscitated by having a tube miraculously put through their esophagus that landed in their trachea. But when we did further scans, there wasn't any airway. So when you look at this image here, can you guys see the reconstructed airway? The breathing tube is everything that you see from the mouth to where it branches off into the two lungs, and there was no other structures available. So in these scenarios, what you ultimately need is, you don't need a tracheal graft, you need a tracheal replacement, and there really is no available tissue source for this. And so we look to regenerative medicine for these types of solutions, and there are certainly many tissue engineered constructs that are already available at the bedside, but the concept of tissue engineering is the incorporation of a scaffold, a cell source, and host signaling in order to recreate an organ that is identical and functional to native tissue. And this area of tracheal replacement actually hit the lay press about 10, 15 years ago with several news-making cases of tracheal replacement that were done for humans. Claudia Castillo was the first human recipient of a tissue-engineered trachea in 2008. And then Andamarian Bayan, he was an Icelandic graduate student. He received a synthetic trachea a few years later, and both of these were published in the New York Times. However, many of these cases were led by a celebrity surgeon named Paolo Macchiarini, and as more of these cases began to arise, there were more and more questions related to the scientific rigor of the work that was being done. And then ultimately, once you're in the lay press, it's not far behind that you make it into pop culture, but tracheal replacement, so you never want your field of study to go viral or become a Netflix special, but Paolo Macchiarini's work is now considered largely untrue, and there's been many offshoots. So whenever one of these series comes out, I get probably 20 to 30 text messages of screenshots of Netflix asking if I watched the special. But I think what ultimately we need to take away from this is that many human lives were affected adversely from this. And we needed to take a back to the bench approach in that regardless of the path that led this to, popularity within social media or within the lay press, we need to understand that there still is a significant problem. And the fact that it was, we need to look at the silver lining in that there still is a lot of work to be done. So our tissue engineering program here at Nationwide is the use of a combination of strategies. We use small animal models to elucidate the mechanisms of airway regeneration, large animal models to study the clinical performance, and then computational models to help us design and iterate additional grafts. And our hope is that in a decade we may have something that can serve as an off-the-shelf solution for these children and adults for reconstruction. So our back to basics approach went really down to the cellular level, right? Understanding that the trachea is far more than a tube. It is a very complex organ that's composed of cartilage, epithelial cells, and its job isn't just to serve as a conduit for air, but to sweep mucus and clear and serve as a barrier for all these pathogens that we are inhaling on a regular basis. And so much of our performance of our tracheal grafts rely upon what we are seeing these stem cells doing. And in this case, we've prioritized the airway basal cell. Now, the airway basal cell is the tissue-specific stem cell of the trachea, and it gives rise to all these other differentiated cell types. And you can see here that when these other differentiated cell types go wrong, these are implications that occur in all these other lung diseases. So I think that there's our improved understanding of basal cell regeneration will also help us in the management of these other disease entities. And then to cite Woody Hayes and his famous Ohio State football coach and his thoughts on passing the football. He says when you pass the football, 3 things can happen and 2 of them are bad. And that's kind of what happens when you create an injury to the airway. 3 things can happen and 2 of them are bad. So basically, if you injure the epithelium, which can happen with intubation, it can happen with our surgery. I don't have a big enough ego to realize that whenever I do an airway surgery, I'm causing some damage before I cause improvement. That 3 things can happen and that could either regenerate, so that basically means that the cell population returns to normal. It can repair, which means that the cell frequency is not quite normal, but you still got normal function, or it can remodel and cause a lot of fibrosis where things they don't look normal and they don't act normal. So ultimately, our strategy within our center is to identify a scaffold that will promote regeneration and we can suppress repair and remodeling. But first, we had to set the story straight. So in the context of airway basal cells, we studied the performance of some of the grafts that had been used in humans. And we really found that when it comes to differentiated cell types that we would want in an ideal trachea, once you implant a synthetic tracheal scaffolds, they really don't behave in terms of supporting normal tissue regeneration. So we had to go back to the drawing board on how do we create a new scaffold. So we understand from prior literature that the trachea is not like other organs because it's not uniformly immunogenic. So I think when you think about other tissue engineered constructs where you're decellularizing skin or small intestine submucosa in order to create a acellular scaffold, you don't need to do that with trachea because the chondrocytes within cartilage are embedded within a dense extracellular matrix that do not elicit any immunogenicity. So we can achieve the same goal of creating something that's non-immunogenic and biocompatible through the act of partial decellularization, where we're using detergents to remove just the top layers of the trachea while preserving the cartilage underneath, which preserves the mechanical support and allows for the trachea to remain open. So we have demonstrated this through a number of publications that we're able to preserve the extracellular matrix, we're able to preserve the donor chondrocytes, we're able to retain graft patency in vivo, and this is with a mouse microsurgical model where we've done probably over 1,000 tracheal transplants. And then we've also, most importantly, identified that they are non-immunogenic. So when you take a donor trachea from a mouse that's not syngeneic, genetically different from another mouse, and you decellularize the scaffold, you put it into another mouse, it's not going to elicit any T cell immunogenicity, so that's important. So we published this, our latest paper in Nature Regenerative Medicine confirmed this, and this is just a slide that illustrates, in the native trachea, you have this beautiful ciliated epithelium, a lot of cells along the basement membrane. After we partially decellularize, there's no cells, and then at 1 month, and then at 6 months. So you have regeneration and preservation of the epithelium after that time. And then more importantly, looking at the epithelium, looking at the airway basal cells is just one cell of many. So we did some single-cell RNA sequencing where we dissociated all the cell types from our graft. And when you take a native trachea and you take all the cells out, you will identify 26 unique cell populations. When you take a syngeneic surgical control, you also find those 26 unique cell populations. And we found that our graft is able to support regeneration of a scaffold with those same 26 unique cell types. So we're also able to restore the heterogeneity of these cell populations following implantation. But as we all know, what you find in a mouse is not the same thing that you can get out of a human. And I think that this is where we're standing on the edge of right now, is trying to figure out a way that we can scale our technology from the mouse to the human. And so this is where we look to creating a bioreactor. Now, bioreactors are all over the place, and they can be used for a lot of different purposes. They can be used to recapitulate an in vivo environment. They can be used for cell culture. They can be used for decellularization. And for us, we were left with this balance that we're trying to preserve, where we wanted to remove all of the cell types that we don't want, while we want to preserve all the cell types that we want to preserve. And when you're in that situation, there's certain limitations of what you can achieve at the bench. So through the support of the Office of Tech Commercialization, we designed a modular adjustable bioreactor for decellularization and cell seeding, and we've been able to demonstrate that we can replicate this process that we have achieved in mice on human scale tissue. And so what I mean by human scale tissue is that we have two surrogates for this, right? If we want to look at the human adult trachea, we use a lamb model, and then if we have a New Zealand white rabbit model that replicates the neonatal airway. So these are the two models that we use to look at the different dimensions of the pediatric versus adult airway. And through this, we can create two distinct environments, one that basically promotes the viability of the chondrocytes, and one that allows us to remove the cells from the epithelium and the endothelial cells of the submucosa, with the hopes of optimizing the cell populations and preserving the viability of the cell populations that we want to stay viable in the graft, meaning the chondrocytes. And so this is a prototype that we developed with the support of the office, in which you can see that our trachea graft is inside of a culture-rich condition, and then we have decellularization agents that are able to go through the lumen of the trachea. And the additional thing is that in order to understand if our graft is doing good or bad, we kind of had to create new goalposts, because there isn't a benchmark for partial decellularization. And so we kind of took to the orthopedic literature. There is some work out there with osteochondral allografts where cartilage preservation is critical, and cobbling together different outcome metrics, we have established basic ways of testing when our graft is going to be ready for in vivo testing, mechanical testing, and then hopefully someday human testing. So preliminarily, we've been able to demonstrate that our bioreactor is able to improve the viability of the donor cartilage cells far superior to our conventional methods of partial decellularization. And what you see here is what we call a live dead assay. So all the red cells are bad, they're all dead, and all the green cells are are living, and they have an intact cell membrane. So basically, A here shows you this is a normal cartilage ring before any treatment. When we conventionally partial decellularize a rabbit trachea, unfortunately, even because of the process that's needed to remove the entire epithelium, there's no living chondrocytes left. But with the bioreactor, we're able to improve this viability substantially. So our ongoing work is really to optimize the conditions of this, right? Because there's either there's certain flow parameters, there's different reagents that can be used. So through the support of the Office of Tech Commercialization, we're hoping to optimize this device so that we can create something that can be used for clinical approaches. So. In conclusion, our goals are ultimately to optimize the bioreactor conditions for partial decellularization. I think that outside of the bioreactor, once we have a viable graft, there's opportunities for commercialization from that regard. And then, our concepts that we are demonstrating some potential in the world of cartilage in general may have applications in other places, even beyond the head and neck. So I just want to thank, again, you all for the opportunity to present, and I'm happy to take any questions.
Thanks very much for that great presentation. Did I understand you correctly that you can decellularize a trachea and transplant that, and you get regeneration of all 26 cell types?
Yes. Currently, that's all in our mouse work. So we can strip all the cells out of the trachea, sparing the chondrocytes, and we implant them. They live indefinitely.
It's absolutely incredible.
They die of old age.
Do you think that's a function of regeneration of the trachea that started, or is that a function of the host factors that support it?
So we're not seeding with any cells. So all of the neo-tissue, so cells that migrate onto the graft, are host-derived. But I think where we are novel is that our process creates an extracellular matrix scaffold that has an affinity for that host-derived infiltration. We weren't seeing any of that even when we were more aggressive with decellularization, we didn't see that. When we used synthetic scaffolds, we didn't see that. So that's what's exciting is that there is this framework that the body has an affinity to migrate onto. And I think that we need to identify how we can accelerate that, because there is a scaling issue, right? Again, this is happening, really well in mice. Our survival following surgery is about 100%, but in order to do that in a rabbit or even do that in a lamb, and then the goal would be a human, I think is still that gap, and that's what we're hoping this technology will help us bridge.
That's awesome. And what are you imagining the transplant start? Who would be the donors in your future product?
So that opens up a whole opportunity for even xenogen transplants, right? Because there's no reason why a sheep trachea, if you decellularize it and it's non-immunogenic, can't be used for just airway reconstruction. And is our thought as a surgeon going to change because right now when you think about an airway reconstruction, you're like, I have to make this big incision. The most morbidity is honestly in their chest incision. There's risk of pneumothorax. There's so much pain related to it. But does that change our threshold for doing a reconstructive surgery if we can just take something out of the freezer and put it right in? So that's what we're hoping to achieve. So,
I have a question. Tendi, that's pretty exciting work you're doing. I had a question about the revascularization of the graft in the mouse model versus what would happen in the human, because in either case you're really mobilizing the entire organ and the whole posterior blood supply is being disconnected, right? Yep. So do you think what you learned in the mouse would be different, obviously, from the human case? So in terms of the actual surgical procedure, how would you navigate, replacing the human trachea?
Right. So I think that that's a great question. So neovascularization, there's a ton of articles even written about this, is one of the major barriers of segmental tracheal replacement. When you take out a huge chunk of the airway, you can't just rely on the ends to populate, enough nutrients for that graft to survive. So just hot off the presses, we're doing some perfusion studies where we demonstrate that after about a month, the graft actually, where there's no endothelial cells and no vessels, we have regeneration of blood vessels in the graft at a month. But the perfusion is something that's lacking. So that's where you can look at bioactive molecules. Composite tracheal grafts. Our NIH grant is basically the combination of, because I think that something that has the affinity that's biologically derived combined with something that is a biomaterial-based bioactive, either a stent or a splint or something external to maintain mechanical properties, I think is ultimately where we're going to live. So, but yeah, so there's a couple of ways that you can think about doing that. You can do a heterotopic implant where you pre-laminate or pre-vascularize. Jan Ranks does that with the New Zealand white rabbit. That's why we do that in the rabbit model, because there's a lateral thoracic artery fascial flap that we've done probably about 15, 20 surgeries, and we've shown that we can get vessel regeneration if you heterotopically pre-laminate under that flap. And you can do a similar flap in humans, and then you can do a microvascular reanastomosis. And then I think that, so I think that where we're going to live is there will probably be a stage procedure ultimately that will lead to tracheal transplant, but probably not for airway recon. So if you have like a patch tracheoplasty model, or if you have just a partial defect of the trachea, those I think can be directly implanted, especially if you're able to tune those properties of your biomaterial in order to really accelerate neovascularization in the region. So,
Thank you very much.
yep, thanks.
It is my pleasure to introduce our next speaker, Dr. Misam Naomi Kararudi, out of our Center for Childhood Cancer. He is a principal investigator and is going to be talking about today a novel mechanism for correction of heterozygous autologous mutations called Remedy.
Good afternoon, everyone. Thank you for coming here, being online. It's a pleasure to be here. Like always, it's fun to talk to entrepreneurs, companies, industry partners, because I think that's the bridge that we need, and the OTC makes that bridge for us. So the topic of my talk is Remedy corrects heterozygous mutation easier than ever. As an investigator here at Nationwide Children's Hospital, my long-term dream is to bring cell and gene therapies to low middle income countries. It means making more accessible with lower cost, and make it to any child anywhere in the world. That means that you have to decrease the cost of manufacturing, decrease the cost of the making these corrections, or even making the reagents available. So primarily my lab and my team focuses on that concept. And we use CRISPR technology as a tool to achieve that. For those who are not familiar with the CRISPR technology, is basically a immune system from bacteria that being adapted to genome editing in human. It consists of a Cas9, which is a protein, can be guided using a single-stranded RNA that you tell them that to complex to each other and makes this Cas9 complex to go and find the genes that you want to manipulate, you want to turn off or turn on. Pay attention to this PAM sequence here. I'll go just back a little bit. Just remember this. I need to talk a little bit more about it in a few minutes. But the idea is that when you design the CRISPR technology and this guide RNA, it just screens the entire genome and finds the PAM sequence and then binds to it and then opens up the double-stranded DNA and binds to specific sequences and nucleotides that you told them to bind to. And that activates the protein that's called Cas9. And then when it gets activated, it makes the cut in both strands. And now you have a double-stranded DNA that's been opened up, but it needs to be fixed. The repair machinery here always makes mistakes. Like if you had a T before, now it is an A, and so on. So the results of this will be a knockout of the downstream genes. there, so it doesn't plate very well. But when you have this mistakes here, it just turns it off. In my laboratory, we use this technology to make off-the-shelf CAR T cells. I don't want to talk about this technology, but those small little things you see, they are off-the-shelf CAR T cells that they are attacking the cancer cells that are green. And you see how they get crazy, and that's the concept that we use CRISPR to turn on and off some genes in the laboratory for cancer treatment or even autoimmune disorders. I will be happy to talk about it later offline, but this is one of the things that we use CRISPR for. But as my role as a director of the CRISPR genome editing facility here at Nationwide Children's Hospital, I have to help people to use CRISPR and genome editing for a variety of applications. So one of the things was neuromuscular disorders that we are pretty famous for here. So I went to the Dr. Mendel, and I said, hi, Dr. Mendel. He said, hi. I said, how are you? He said, okay, good. And then he was like, I said, I want to use CRISPR for neuromuscular disorder. Good for you. I'm sorry. He was really, really happy that I came with that idea because he'd been waiting for it, and he had some idea to use it for. So I said, okay, which disease you want to target? And it took us a month with 6 of these individuals that they are very famous in the neuromuscular disorders to pick up a single disease that there is no treatment for, and CRISPR potentially could be used for them. And after a couple of months, Dr. Shahang and Dr. Mendel called me in their office, said that we found a disease for you. And that was IBMPFD with a heterozygous mutation in a gene called VCP. Let me give you a little bit of background here. So in every single gene in our body, we have one allele that comes from mom, and the other allele comes from dad. So it should be 50% and 50%. And when you have a mutation in one of those alleles, it can be it's disease-causing mutation, and that is called heterozygous mutation. I'm just simplifying to not make it so complicated. But remember that it is 50%, 50%. And if you sequence it, you clearly see 2 peaks here that one is, let's say, it had to be C and the other one is T, while it has to be something else, okay? And then if that happens, you get this disease here shown in the picture. If you remember, I told you that CRISPR can kind of screen the entire gene, and as soon as it finds its target, it makes the cut, right? But it cannot distinguish very well if there is only one single nucleotide here difference. So C became T, and that's not going to be very precise because then it cuts the wild type or healthy gene and the mutant gene, right? So you just get rid of both genes and you don't need to do it. So Dr. Mendel and Dr. Shine, instead of helping me, they just put something else in my plate that I had to solve. So I went back. If you remember PAM sequence, I told you I thought that biology of CRISPR, that the PAM sequence is essential. I thought that what if we put that or design the guide RNA in order to include that single nucleotide mutation in PAM sequence or close by, like in the close vicinity, maybe in that case, CRISPR makes mistakes and doesn't go and targets the wild type, it only targets the mutant allele. And that happened. If you see here, the healthy allele stayed as it is, and the mutant allele get targeted and it couldn't be detected. We were pretty happy. We were actually turning around because no one had described this before in this setting, and we could now turn off a bad gene and let the good gene to work. And if you pay attention, this guy has the same hairstyle as mine. Please don't laugh. So we said, okay, now we have an in PAM or close to PAM CRISPR strategy, the strategy that we could use for other heterozygous mutations. We talked to Dr. Alison Bradbury that she works on a disease called TBCD, 50% wild type, 50% mutant, same concept. If you sequence it, you clearly can see it. We said, let's now use our technology one more time and then we can get rid of this mutant allele, and we ended up having 50% wild type. It happened. But then Yasemin, my lab manager, called me and said, I think we have a sequencing error here. I'm sequencing 64% healthy allele. I don't know how is that possible. I said, are you sure? And then she said, yeah, I'm sure. I said, let's sequence it again. And we sequenced it again, and it was again 64%. And then I asked her, did you check Dr. Mendel's data? She said, let's do and check again. And that was 92% healthy allele. We were like, how is that even possible? So we spent month and month to study what was the reason. And the reason that surprises us the most was if it happens only if you provide a extra exogenous donor DNA template to be used for the correction. You say that, hey, cell, I knocked you out, use this one to correct yourself. Well, we didn't do that. And that means that you have to provide that template using AAVs, DNAs, or other stuff, and that costs you $1 or $2 million more to treat these patients. And we didn't do that. And you remember I told you my job is to decrease the cost and accessibility, and that was like, so how did that happen? And that's how it happened.
Your cells contain chromosomes from each of your parents. These chromosomes carry different versions of the same genes. Sometimes when a person inherits a healthy gene from one parent and a mutated gene from another parent, they develop a genetic disorder. Remedy is a new gene editing approach that can help cells repair mutated genes in cases where the cell contains both a healthy copy and a mutated copy. Remedy uses CRISPR, a gene editing tool to cut out the mutated gene. CRISPR is very targeted and only cuts the specific sequence the researchers tell it to cut. After CRISPR cuts out the mutated gene, the chromosomes move closer together and begin to communicate. The cell's natural repair machinery steps in and uses the healthy gene as a template to repair the gene that was cut. The result is that the cell will have the original healthy gene on the chromosome from one parent and a corrected gene on the chromosome from the other parent.
So that's how it happened. Make the negotiation happens and you can solve a lot of problems in the world. You have a good copy, you have a bad copy. You tell them like, don't work, and then this one comes and starts talking to each other. And that's how Remedy works. You are using already existing good copy of the gene in your cells to correct the other genes that you wanted instead of paying millions of dollars to correct that mutation. Then we thought that if that is the case, if we add some drugs that can kind of stimulate and enhance these processes, maybe we even can get better correction. So Genesis in my lab did test a lot of different drugs, and that's 64% went to 82%. And we thought that maybe we can do it in other diseases as well. And I wanted just to be kind of cautious and give it to other persons. So I asked Sarah Ram and Dr. Reynolds that, can you correct cystic fibrosis for me using Remedy? So they had cells. I went to them and said that you go ahead, you design, you treat, and then come back to me with your results. 88% correction of cystic fibrosis. And that is the most common genetic diseases affecting the airways. I said, okay, maybe this is just cell-specific. We went to Dr. Juhi and Dr. Roshini with my research scientist, Nushin. She corrected it again. With the very high efficiency of a disease in ITPR, 3 heterozygous mutations. Then we said maybe it's not even enough. We went to Dr. Sad and with Colin, my lab, to correct a mutation that causes like premature aging, and that's called progeria. You see the complete deletion of the mutant gene and correction of the mutant allele using the endogenous healthy allele in the cells. we said that now we have to think a little bit about the delivery systems. So with Dr. Sad, we proposed a methodology to deliver that, and we won the NIH Targeted Challenge for revolutionizing the gene editing for in vivo application for heterozygous diseases. You know that this is not possible without the team, and this is the team I want to really thank for everything they've done and for every single things that we did as a cool lab. Thank you.
Great talk.
Thank you.
I was just wondering if you could expand a little bit more on that delivery piece
Yes.
that you hinted at.
So there's two things that we have to be really careful here. One is what the disease-causing mutation is, where is the most important organ to treat, and then we can just talk around that a little bit more. For example, for the blood disorders, you can just take out the CD34 bone marrow cells, correct them, and put them back. So there is no delivery needed. For in vivo genome editing that we started recently for a seizure-related heterozygous mutation, you can directly put the CRISPR or other reagents using LMPs into the brain. For neuromuscular disorders, with Dr. Shahang's lab and my team, we are working with a company called Metagenomi that brings, just give us a very small cast that we can just fit everything in a single AAV vector, and when we can deliver it easier. So the variety of the ways that you can deliver this system depends on your disease, the organ that you want to target, and how much efficiency you need to achieve.
Hi, thanks for the presentation.
Sure.
It was, yeah, congratulations on
Thank
your
you.
work. It's exciting. As I think about the commercialization of this technology, which it seems clearly you're headed to the clinic, what do you think will be the first application that you'll be able to address at scale? And what might be like one of the biggest commercialization challenges that you're facing as you think about making this a product or, clinical service line that could be deployed globally?
So we heavily invested now on the VCP disease because we have almost every tool available. We have our clinical team ready. We have a historic studies going out now in the hospital. We have the AAV manufacturing ready. And also we have this collaboration with this company that provides the cast for us. So that's what we are doing right now. But as I told you, I'm a PI in a cancer center. So the CD34 genome editing, that is really what I'm interested in to move forward, because the sickle cell disease being approved, and it's the same thing, that you knock out a gene, they didn't have to correct, and now we see that we can correct too. So it's a pretty straightforward way. The FDA is familiar with everything needed to move forward with this. We have the team here, and the cell therapy team that we have here is very open to collaborate. But the other things that I'm very excited about is the seizure-related epilepsy that I'm working on now. And now I just started with a potential collaboration with the University of Oxford to put the remedy directly into the eye. So everything that is topical, local, and you can have control over what is going to happen, it's the things that I'm seeing that to happen in the next few years.
Thank you very much.
Thank you.
At this time, we'll ask our panelists for the day to come up to the stage.
So just real briefly, I'm going to introduce our panelists, then begin a conversation today that's focused on, it has a focus on the MPDC, which is the Midwest Pediatric Device Consortium, but we've invited some other expertise to the panel to help us think through some of this. And just to give everybody a little bit of context, we have limited time today. I want to devote as much of it to discussion as possible. But, from the FDA's website, if you're trying to kind of capture big picture what a PDC or a Pediatric Device Consortium is supposed to be, you've heard referenced a couple times today that there is this paucity of resource and really a lack of translation for med device on the pediatric front. And so the FDA launched a program several years ago. I think it's in its, is it pushing 20 years, gentlemen? Pushing 20 years this program's been around, called the Pediatric Device Consortium. And from their website, to address this health inequity, FDA funds PDC grants program, the PDC grants program to advance medical device development. These nonprofit consortia provide a platform for experienced regulatory, business planning, and device development services, such as intellectual property, advising, prototyping, engineering, laboratory testing, and grant writing to foster achievement, or advancement rather, of I have multifocal contacts in that are not working as well on this phone as I thought that they would, so we're gonna have to remedy that later. Advancement of medical devices for pediatric patients. In addition to key advising services, the consortia also provide critical funding to innovators to direct and advance these projects. So everything but the kitchen sink is described in what these consortiums do, but the reality is we've learned about them and the different PDCs that exist. There were 5 funded in 2023. One of them is here at NCH, the MPDC. And then the Southwest Pediatric Device Consortium, which I'll introduce their members here in a second, are 2 versions of that. But I've found that, each ecosystem has done what it needs to do with the resources around it and kind of custom build out a methodology to try and foster this type of work in the region they are. And they all service nationally the needs of med device developers. And so that's the topic we're going to be talking about today. Let me quickly introduce our panelists. Dr. Corey Chris is an assistant professor of clinical surgery and pediatrics at Nationwide Children's Hospital and the Ohio State University College of Medicine. He's the director of the Nationwide Innovation Center, which you heard about a little bit earlier today, and he's also the co-director of ECMO and associate program director for surgical critical care. Dr. David Ekman is the founding director of the Center for Medical and Engineering Innovation at Ohio State University, and he's vice chair for academic affairs and professor of anesthesiology at the OSU College of Medicine, as well as professor of biomedical engineering at the OSU College of Engineering. And so Dr. Chris and Dr. Ekman together represent the co-PIs of the Midwest Pediatric Device Consortium, Dr. Ekman serving as the CEO and Dr. Chris as the COO of these efforts. As I mentioned earlier today, the Midwest Pediatric Device Consortium, the academic partners are, or listed on the grant anyway. There's multiple partners. We're trying to expand that all the time, but are the Ohio State University, Nationwide Children's Hospital, Cleveland Clinic Children's, and Cincinnati Children's Hospital. Our next panelists today are Dr. Chester Koh. He's a pediatric urologist, and I love this word. He says it's been around a long time. I haven't used it a lot before. Dr. Ekman, this would apply to you, is a physicianeer at Texas Children's Hospital and a professor of urology, pediatrics, and OBGYN at Baylor College of Medicine. And then joining him from the Southwest Pediatric Device Consortium is Professor Bala Haridas, and he is a professor of practice, Kane Faculty Fellow, and the Director of Translational and Industrial Research in the Department of Biomedical Engineering at Texas A&M University. And so, Dr. Haridas serves as the Deputy Executive Director, and Dr. Koh serves as the Executive Director of the Southwest Pediatric Device Consortium, which includes Texas Children's Hospital, Baylor College of Medicine, Texas A&M, Rice University of Houston, and University of Minnesota. So those are our 4 PDC panelists today. And then our final panelist, last but not least, is Eddie Pauline. Many of you in the room know Eddie. He is the president and CEO of the Ohio Life Sciences Association, which is our trade organization for life science in Ohio. And, OLS is one of the pieces of Ohio infrastructure that stepped up and leaned in to become a part of the MPDC and make it possible. There are other organizations, ESPs like Rev1, Cincy Tech, Jumpstart, Jobs Ohio, that are in the room today that also served a really critical part of getting this off the ground. So that's kind of the background and context. So maybe to start things off, one of the first questions I'd like to ask, maybe focused initially on Corey and David, we heard a really broad description of PDCs. How did you structure the MPDC, and maybe why did you structure it that way, and for what stakeholders were you trying to address by doing that?
Sure, I can take this one. So, as Matt said, all the PDCs are structured a little bit differently. One of the really unique focuses that we wanted to rely on here was collaboration and customer focused. So we wanted to be collaborative, and that involves, all four of the institutions that Matt talked about: Nationwide, Ohio State, various schools at Ohio State, Cincinnati Children's, and Cleveland Clinic Innovations. And so we wanted to be collaborative from that perspective. And then we really wanted to create an organizational structure that was focused on the customer being pediatric innovators, device companies that can come to us and we can provide the necessary services, not necessarily a one-size-fits-all model, outside of the funding, which is, one of the other major PDC objectives is we actually provide funding through a competition. But outside of that, really providing those resources, and the structure is really, we have an executive committee that oversees all of the strategy and our objectives, and really looking forward to overall strategy. And then we have our program manager who manages day-to-day operations, McKay Knoll, who's here. And then we have a departmental structure that is focused on all elements of those four institutions, being engineering and prototyping, regulatory, outreach, real-world evidence, bioinformatics. And so the thought is that we highlighted the expertise of those four institutions. That being said, one of the other important points, to Matt's comment is that we also really want to lean in and rely on the expertise of everyone who's partnered with us as well outside of those four institutions. Involved in this grant, obviously, Wayne Embry from Rev1, Bill Dunleavy from Ohio Life Sciences has been really instrumental. That being said, we know that we can't do everything internally, so we also are developing a network throughout the entire state who can really lean in and jump in to help those companies get where they need to go. Because, we don't want to, we can't do it all, and so we're relying on the expertise of Ohio and the Midwest to do that. So you start there. David, if you want to add in anything.
No, I think, Corey, that the way we envision our function is really as helpmates to companies who want to grow and who want to place products through the FDA regulatory process into the marketplace. And that has many, many points at which failure can occur and help is needed, especially for small businesses.
And then I'm curious, Chester and Bala, are you structured similarly at the Southwest PDC?
Yes, so we're a multi-institutional consortium, as you mentioned, the four other institutions besides Baylor College of Medicine and Texas Children's. And I think the key lesson that we've learned is that pediatric device development is challenged. I think Dr. Cadado did a great job of summarizing that in the slides. But I think the solution to that is we have to de-risk further within academia and children's hospitals more than for adult devices. So I think that's why the PDC is a great example of having that ability to do that, but it does take funding. And going back to the story of the program being around for 20 years, I mean, it's really an act of Congress that led to this back in 2007. The Marion County Pediatrics, which has a very prominent voice on the Hill, working with all their stakeholders, have really pushed that act of Congress, as well as the reauthorization every 5 years, as well as the annual funding that allows us to do that. So this really adds funding to work within academia and children's hospitals to provide the funding to innovators to have perhaps seed funding for their projects, for regulatory help and reimbursement help and other consulting help that we can provide the funding for. So it's really a needed area because we are a small population in terms of who we serve as well as low profit potential, but still we need those devices. It's really mission-based work that we're doing, but still very important because our kids need it. Paul, anything else you want to add?
Sure. I mean, having done this since 2018, Chester and I started working in 2015 together. One thing I realized was there's a period of time in which you have to try and figure out, how you fit into the regional ecosystem, and it takes a couple of years for that to shake out. For us, it took us that much time, and slowly we realized that we have really sort of an internal innovation portfolio, meaning our own internal physicians, partners from the different children's hospitals, but primarily Texas Children's, who have ideas. So you have to find a pipeline for those. But then there are these external innovation partners who are largely, startup companies that are solely looking at pediatric device, or maybe companies that are looking to expand their labeling for pediatric indications and so on. So you have to find a way to really fit them into your portfolio and the pipeline of things you do. So it does take a year or two to kind of let that shake out as well. So that was our learning.
That's really helpful. Thank you. And I want to play off of something you just said. You talked about how the ecosystem kind of had to learn your consortium, and you had to learn the ecosystem and get connected on that front. So we're wanting to model after that and be a repeat customer, if you will, of this program from the FDA. And so thinking about that, maybe this next question directed to both either Corey or David, but then also Eddie adding on to this. From the investigator perspective, what gave you confidence that Ohio was a good ecosystem to tackle this? And then maybe complementing that, Eddie, what has Ohio done to prepare itself to be ready to service something like this? And maybe what are we doing to continue to support it?
Well, I think it was very evident to both Corey and me that we had a tremendous geographical and institutional advantage. You just look at the map of Ohio and say, okay, here's Cleveland, huge metropolitan area, Columbus, huge metropolitan area, Cincinnati, large metropolitan, very large metropolitan area, and each of them populated by a world-class children's hospital. And I don't think you can find another region of the United States of America that has this kind of density of pediatric care at this level of excellence. Then add to that, right in the center, the Ohio State University, a world-class research university with an engineering college that has about 500 faculty. The institution as well has a world-class medical center. And so with a faculty of over 1,500 physicians, we have, I think, all of the resources in the academic posturing in order to not only compete, but to be successful in this effort. Couple that with changes in the state that emanate from legislation, that emanate from a drive in Ohio to, in some ways, reinvent itself technologically. I think it was just a matter of this is the right place at the right time, and we recognized that, and that gave us a tremendous amount of courage that we would be successful.
Well, Matt, first and foremost, thanks for organizing another great day to showcase your tech. Your team does a great job. All of the innovators in the room should be really proud of the pipeline of innovation coming out of this institution. It makes our job as the association a lot more easier when we're bragging about this institution across the country. I think what this was for me is a really interesting observation of how to get our distributed network of life science hubs to play nice with each other. It is hard to get Cleveland, Columbus, Cincinnati to collaborate on a lot of things. It's even harder to get the institutions within those cities to collaborate. So the fact that this opportunity brought everyone together in a nice, concise, powerful proposal is really something to celebrate. And I think it gives us a model of how to do business and how to go after even bigger opportunities down the road. This also gives us an interesting opportunity to continue to help educate people about the expertise that we have in the state. I just spent 2 days in Washington, D.C., meeting with some members of the Ohio delegation and other partners. We have a lot of work to do to help people understand here in Ohio and outside of Ohio of the true expertise, the capabilities, the unique and superior assets, however you want to describe it, that exist in this state. We need to educate our elected leaders about those assets because they're critical in driving more of these types of federally funding opportunities our way. So to have one here now and to be able to prove that we can make something of it, I think will only feed that bigger opportunity to get those DC-based people excited about what more is possible in Ohio. So it's a really exciting opportunity, well-deserved opportunity, and it's going to inform, I hope, how we do business as a state going forward.
Great. Thanks, Eddie. This next question I'll direct first to Chester and Bala, but really for the group. One of the things that I was really encouraged by, but also kind of surprised me when I first started getting the opportunity from Corey to come to some of these meetings and learn how the PDCs operate, was that the FDA wasn't just a funder in this process. This isn't we're going to throw $7 million for the next 5 years over the wall and come back to us and tell us how you're doing later. But it feels like there's a lot of interaction with them. And so I'm curious if you can comment on how does the FDA participate in this program? And if you're a company approaching a PDC, what can you expect from an engagement perspective with the regulatory body through the PDC?
Yeah, and that's a great question. I think some of you may have heard the phrase that this is a new improved FDA, and I think many of us have seen that over the past 10, 15 years. We found them to be more responsive, especially for pediatric devices, and there are really some key individuals at the FDA now who are really our program officers, both Dr. Vasoom Paris, who is a Chief Pediatrics Officer, CDRH, as well as our Pediatric Device Consortia Program Director, Erica Torgerson. So both are very available to us, but also really making changes internally such that, one complaint we heard or one comment we heard before was that a pediatric innovator would have their device up for review and they would get these responses that seems like the reviewer didn't quite understand pediatric devices. They're a smaller market, lower profit potential, and so hence they were not quite successful. But nowadays, they're trying internally to make sure that there's at least pediatric expertise in the room so you don't face those challenges in the review process. So those are some of the things that we have been seeing over the years. I think one thing the PDC program can do is something called the PCI meetings. And the PCI meetings stand for the PDC, the Chief Pediatrics Officer of CDRH, as well as the innovator. And that's something only the PDC can really request, where an innovator gets about a 50-minute meeting, then you need to prepare. You need to come up with a few questions and prepare those questions in advance so that you get time with Dr. Paris and Dr. Torgerson to talk about 30,000-foot level about your regulatory strategy, whether that seems to be the right one for you. Can you imagine that probably didn't exist before, but that's available now, and that's free, and we can help support the regulatory support in order to get to those meetings. So these type of things probably didn't exist before, but I think that's some of the ways that more availability is there to help those pediatric devices move forward.
Yeah, my experience has been, I've been in front of the FDA so many times in my career. By the way, full disclosure, I'm an Ohioan, so just for the folks who know me, lived here for 25 years in Cincinnati. I moved to Texas 10 years ago, so before that, 25 years at Cincinnati as faculty at the University of Cincinnati and in the local industry, med tech industry there. But back to the thing. So my, the FDA program manager and the chief pediatrics officer, they're fully engaged. That's been my experience is, a meeting with them, we have quarterly updates with them, and they get down into the details of what we're trying to work out, and they are sincerely want to help us and see us succeed, and also keep us focused. And their oftentimes they'll say, hey, that doesn't fall within the mission of what we're trying to do, and that type of feedback is useful, so you don't waste time going down a certain path in terms of initiating a new project with a company, etc. So they're careful in telling us what works and what doesn't as well. So that's been my experience, and it'll be a very positive one for you folks as well, I'm pretty sure.
I'll make a quick comment about collaboration across PDCs, because obviously we're new to this, but Chester and Bala can speak to the historical requirements and what's been done in the past, but there is more of a reliance this cycle on finding ways to collaborate with other PDCs. I think, we spent the morning together and talked about ways that our institutions do things a little differently and how projects we can collaborate on. But realistically, the FDA has mandated collaboration across PDCs, and that involves things like combining funding for one project so all PDCs can come together and put their funding towards one. There's more unique funding structures where you can fund the same company and this by multiple PDCs, but not on the same project. So there's some more, leniency there. But, the other thing that we talk about is also sharing deal flow. Companies that come through each PDC, discussing that amongst the PDCs, because a lot of them are regionally focused. So how do we share that information, and how do we optimize the resources? Maybe there's a company that we can collaborate on together to get to the next steps. And so we're focused on that, and that's one of the reasons why we're really excited to actually have them here today and discuss everything. So yeah, we're excited to work with them.
I think we found that FDA is very open and transparent with us in this program, which is a tremendous benefit to us. We can go to them with questions. They also provide us a great deal of guidance. I'm sure that's true for all of the PDCs. They're very sensitive to the needs of the companies. Their real goal is to get pediatric devices into the end users. One of the things they suggested to us in our process of negotiation with them for this program was that institutional review boards, for instance, may have a lot of expertise in helping to design and implement and approve clinical trials for small molecules and for other types of medical interventions, but may have very limited experience in dealing with medical devices. And therefore, they suggested to us, and we implemented a program by which the MPDC can provide engineering consultant support for IRBs. So a product in development that may not go through one of our core institutions for its clinical trials. Wherever it does go through a clinical trial, needs to get IRB approval, and that IRB can draw on support from the MPDC.
So just to add, I think the consortia program, each of us do it differently, but I do think there's something for everyone here. I'm a physician. I'd like to see more physicians get involved, more surgeons involved in pediatric device development. I call that clinically friendly research. But there are ways that you can do pediatric device development working with engineers through capstone programs that gets those unmet needs in your head and actually towards a prototype development. I think if you're an innovator, we have the ability to pay for consultants for regulatory and reimbursement work. That's, it's those crucial dollars early in development that you need to spend every dollar wisely. That kind of non-dilutive funding can come to you. So those are some of the things that we have provided over the years that have been crucial to help those pediatric devices move forward.
Great. So that conversation actually ticked off like 2 more questions I was about to ask. So thank you for taking it that direction. But it does lead into kind of this concept that I think it's really important that the audience hears today, and that is, with the PDC, you've heard the panel talk through the different functionalities and the different types of engagement. I think it's a misconception that the only reason you would interact with a PDC is because you need funding, and that funding might be limited if you're dealing with an individual PDC. It's really encouraging to hear that this network of consortiums work together to sometimes co-fund projects, and they can increase that amount a little bit. But I think it's important to understand any company working through the development of a pediatric device could and should engage, not just companies that need funding. And I'm curious for the group on stage today, have you found that there's a particular demographic or certain types of companies so far that have benefited most from that, or a stage of development, or do you deal across the spectrum of stage of development? Is there a sweet spot for the PDC?
Yeah, so I would, just because we're limiting the funding that we can give up to usually $50,000, and we by choice only give $25,000, that really leads toward the PDC starting off mostly with the earlier stage ones. As you get later, you need more and more funding that we're not going to be able to provide. But that's where the partnerships with venture funds, seed funding sources is going to become much more important. And we've learned that here in Ohio, you guys have a great system already. And so that's really one of the leading areas in terms of the amount of funding you have available for later stage funding. But I do think that probably if you're asking what are the best device, what has the most success, and that's really those that get a big enough market. I mean, we're challenged in terms of pediatric devices of being only 0 to the 22nd birthday in terms of what the FDA considers pediatric. That's a small market. So if you happen to have a pediatric device that has adult usage, that's going to be probably the secret sauce in some ways, that if you can find that adult use in addition to pediatric use, but of course we always push pediatric first, have it developed that way, I think that's probably, you're going to have the best chance of survival.
If I may add to that, so one of the key things to keep in mind is how do you activate your ecosystem, right? And so your ecosystem, meaning the academic sites plus the ecosystem partners, which could be incubators and other support firms like design firms, et cetera. Because what eventually you'll come to the point is you'll realize that the tens of thousands of dollars you're able to allocate per project is obviously not sufficient, right? So you need to get to the stage where you're raising the 200, 300K, and then the 2 to 3 million. And so you have to think of what is your portfolio planning strategy for funding. So if an innovator is coming in and saying, hey, I need help, oftentimes we think of, okay, if we're going to take this on, where do they go next after we finish step one with them, right? And so we've learned from some of our successes that you have to think of, okay, which is the ecosystem partner that this particular innovator can move to and write that SBIR grant, phase 1, phase 2. And, so you go from tens of thousands of dollars to a couple of $100,000 for a phase 1, and then you get to the 2 to 3 million, phase 2 grants. So you have to think of which projects will have those legs, because until you do that, you're not going to be in a position to raise any private money. And so that is a very critical thing. The other good thing is there is now, thanks to some of these new big health initiatives at the federal level, like ARPA-H, et cetera, there is money on the table if we think big. So, but by that I mean, can you think of building a team in your consortium or work with some of the other PDCs and go after, say, solving an entire disease in a whole class of patients, right? And talking about device-based solutions. So then you get ready to do these big ARPA-H type grants. So, but first the building blocks have to be in place, and then we, think of, okay, what should your portfolio look like, and how are you going to help these companies once they finish accessing some of the medical device funding that you have, and how do you continue to help them? And really that comes with one extra zero on the dollar side. And that's what it comes to, really, at that point.
Right. And you really got to, I think you guys have done a great job already, really seeing what's already in your local ecosystem and work your way out. I think that's what we have done in terms of the consortium that we started. Luckily, we were in Houston at the time when innovation was on the rise in terms of device innovation, mostly in adult. We just tried to make sure pediatrics are there as well. But I'd say find all the stakeholders who are local, get them together as you're doing, and then move on after that. I think that's been a great game plan for us.
So that makes me, for Eddie, maybe a next question to play off of that. We're talking about, these PDCs, they have a national focus, right? It's everybody can engage. It's not a regionally focused thing. That being said, a lot of the infrastructure that you're referencing in Texas, the infrastructure we've referenced here, many of us do have a conviction about trying to build the ecosystem in Ohio as well. And so I'm wondering, Eddie, if you could speak to, with this effort, even though it's nationally focused, what are the opportunities for Ohio in this to get some state-focused return as well here?
Yeah, I mean, I think, at least from my perspective, this adds some healthy pressure to our ecosystem. I mean, again, we've got the ingredients here. I mean, we've got Reed over there on the design side. Dwayne can help some of these leaders get coached through the process. We have our investors here in the room today. I think what is going to be a challenge is just the size and scope of what our current set of partners are able to provide, which is why this is going to be a test of can we use this to put pressure on the right, maybe broader state resources or industry resources to say, hey, this isn't going to work as well as it could unless we have the right talent in place in the state, unless we have the right funding structures in the state in place, and of course, if we have the right space available for these organizations to grow into. It would be unfortunate if this became another example of Ohio-based IP leaving and going to be developed in other places. So how can we really ensure that if this is going to be an opportunity to draw in opportunities and build opportunities from the ground up, what are we doing as a state to kind of keep that IP here and keep that growth here? We've got lots of examples, some horror stories of great IP leaving, but we are increasingly hearing some stories about IP being developed and staying here. We've got some stories now of Ohio IP being so attractive, then some Boston-based companies are moving here because of it. So this whole opportunity feeds that, but certainly puts more pressure on the ecosystem to figure out how to make it all work.
And if I could add, we've seen a lot of movement from Boston and other crowded areas like Silicon Valley to Texas, and likely to Ohio as well, because as you probably know, if you spend any time in Boston, it's really become a company town with all the work that you see in Kendall Square. The startup space are being squeezed out. So I think it's a great opportunity for you guys to really develop on that this can be the startup area as it has been in Texas for the past 10 years.
Thank you very much. Another question I'd like to ask Corey from kind of the NCH perspective. So I've watched you in the year and a half you've been here have to wear like 30 different hats at different times, and I enjoy conversations we've had about how one activity bolsters the other and back and forth. So can you speak a little bit to, and I would assume this will be true at the other institutions as well, but how has your MPDC experience and being a part of this team impacted your thought process about some of the innovation cycle you're trying to drive at NCH? And do those two intersect? Where do they intersect at?
Yeah, it's a good question, Matt. I think, and we talked about it this morning about where the deal flow comes from. And I think, there's definitely overlap between what I'll say the Nationwide Innovation Center and the MPDC. However, there is sort of the boots on the ground, let's try to mature all of the products here within each institution, for example, NCH, Cincinnati, Cleveland Clinic, and then also tap in intermittently to those PDC resources to catalyze them in a way that they wouldn't be able to do if the MPDC was not here, and actually get them on the stage to talk to the other PDCs. So I think the reality is the PDC being here has launched the local innovation center sort of into the stratosphere for optics and opportunities, and I see them as a stepwise process where we want to mature those in a way that's internal, but also at some point tap into those PDC resources, because there's some stipulations of the PDC. The first thing is the products that get funded have to be mature. So much so that our first funding cycle, you have to be a company. You can't just be an investigator that does not have a company. So you have to be mature. There are some other PDCs that can offer funding for just investors, but it's mainly mature. The other major thing for the stipulation is that for the funding we get through the FDA, we can only 25% of that can be given to internal projects out of the 4 institutions. So there is an incentive to fund projects outside of the 4 contributing members, OSU, NCH, Cleveland Clinic, Cincinnati. So to offset that, we really want to drive that at each institution, that can sort of offset that lack of direct device funding. So is that?
Wonderful. Thank you very much. We're on a little bit of borrowed time, and we are the last thing standing between people and libations and good food. So I do want, though, before we conclude, to give the panel everybody on this is for everyone, if there's a last, final thought, comment, something that if you wanted to make sure everybody in the audience got a takeaway from today's panel, or just a final thought, what would it be? No requisite to do this. Just if there's something we didn't touch on today that you'd like to do, please feel free to offer it.
Sure. Well, my parting thought would be that the MPDC has a lot more to offer than financial resources, and that includes help with clinical trials design, with manufacturing, for prototyping, with regulatory management, et cetera, that goes far beyond what the dollar awards can mean. And there may be partnering with businesses that is non-financial but equally valuable.
Just back to the idea of leveraging this opportunity to drive more resources to the region, to the state. I'll go back to my visit in DC yesterday. We were with part of a fly-in with AdvaMed, which is a national med tech lobbying group. Former Senator Richard Burr from North Carolina came in and spoke to the crowd before we went to Capitol Hill, and reminded us that it's a very interesting time in politics right now. And as you're talking, he specifically talked about the fact, and I think this applies to the state as well, never has there been a time when there's been so much newness in our collective elected bodies. A lot of new faces, a lot of institutional knowledge has left those elected roles. So there's a lot of newness, and the appetite to learn and understand more is not where it used to be. So with that type of audience in mind, how do you work to educate them on what something like this really is, to get them excited about it and help them realize the bigger opportunity that it could create, not only economically, but in the development of life-saving treatments. As I was listening to the presentations today, this stuff is complex, and having someone take a real complex topic and, pardon my friend, dumb it down in order to try to drive more investment is a really interesting skill. So I think collectively we need to think about how to articulate the value here in a succinct way to that broader audience, particularly elected officials.
So I'll say 2 things. One is this particular program gives you the opportunity to do what I call two-way leveraging. As much as possible, marketing the fact that you're there will enable researchers in other STEM fields, engineering, et cetera, say, hey, I'm writing this huge grant, can the PDC help me by finding a clinical partner, right? So you talked about the soft connections, that it's really important, and we've been, surprised by the amount of leverage we provide folks who are writing grants in certain areas and have a way to focus on pediatrics to make them more fundable. The other is you're going to have fun doing this, so this is going to be a fun ride. That's my personal, opinion. So enjoy it.
Right, and then my message probably is being a physician, trying to make sure that physicians stay involved in this, and so be sure to reach out to them. I think many of us don't know that this opportunity exists, so be sure to get that out, but also ability to work with engineers. So that's really from the physician standpoint. I mean, I call this clinically friendly research, maybe a solution to burnout, give you something to work on. But also for the innovator as well, because we want to help you to get into the hospitals, to work with the physicians, and what is usually a volunteer experience for the surgeon or the clinician to work with innovators to do the clinical study. We've added money to that with faculty seat grants. So those are things that can occur to help put some money to the system to help drive those partnerships of engineers and innovators with physicians to get those devices forward. So.
Great. Well, thank you very much to our panel. How about a round of applause? We are at the conclusion of our showcase for the day. I want to thank you all again for being a part of this. Our sincere hope is that each year, or even if we start doing it more often, if we have events like this, that you do want to come and you want to see a little bit what we've got in our showcase and what we can put on the agenda. But I think equally important, one of the things I was doing as I was sitting over here today, kind of looking around the room, I see Cincy and Columbus investors sitting by each other and talking to each other. And I see these different groups from the different regions and different functionalities within the state commingling and gathering and innovating with kind of our thought processes around how we're approaching things. And so I sincerely hope this venue and this event for you has started to become something like that too. Like, I want to go to this event because I know there's going to be enough people there. I'm going to meet somebody new. I'm going to talk to somebody about an effort I wasn't aware of. We've tried to populate tonight's reception with a number of people that can certainly talk to you about Pediatric Device Consortium, but there's a lot of other interesting things in the room and interesting things going on. All of the inventors that were here, with the exception of Dean, that we're here presenting today are going to be here for the networking reception afterwards. I would encourage you to stay, meet a new person, talk about a new initiative, create a follow-up. Thanks for coming. We're glad you were here. Have a great day.